I should be turned. There we are. Happy days. Technical failure already. Right, good morning, ladies and gentlemen, and boys and girls. You're filing your way in, find a space, and see what's in front of you. There are screens on the outside and also on the inside here, and if you want, you can come a little bit closer. Nothing's going to jump out at you, I promise, other than Jess, maybe. So, I am Ben. I'm Professor of Evolutionary Biology at the University of East Anglia. I've got Dr. Jess French here next to me, who is a veterinary surgeon. Catherine as well, who also works at the university and has pretty much made today happen. She's our star today. Got Ellen here and Emily. So we've got a whole bunch of us, and as you've probably seen already, there's a big bunch of dead stuff in front of you. The first thing that I need to mention is tell you the species. So we've got two very different animals in front of us here. We've got a beautiful big Jaguar lying here next to me and a slightly smaller cat over here. It's not a small Jaguar, this is an Ocelot. Now both of these animals are found in Central and Southeast Asia. The first thing that I want to get across today is that everyone on this side of the barrier here absolutely loves animals. Everyone loves animals? Yep. Anyone doesn't like animals? No? Yeah, okay. These guys as well. We all love animals. It's very sad that these animals are dead. I'm going to make that very clear. We have not killed these animals for today's activity. Both of these animals are from British zoos. There's a big debate as to whether you like zoos or not. Some people love zoos, some people hate zoos. Some people are in the middle somewhere. However, these animals are part of zoological collections in the UK. They have both been euthanized because the decision was made they weren't very well anymore. And legally, in the UK, zoo animals like this have to have a dissection in order to see why they've died, what we can do as a biology community to make sure their welfare, the way we look after them, is maintained. So Jess's job, her actual job today, is to go through this little Ocelot and see if we can find out the cause of death. Now, we know that they were both euthanized. This one's already had its post-mortem, so this is just for show and to show you some of the external anatomy today. We will be going through this small Ocelot to see if we can learn anything from the animal, to see why it died. Was there an issue with captivity? Was there a disease? Was there something that we don't quite know what was going on inside? So this is actual work that we are doing as biologists and vets and others within our community. We do also have some of our colleagues from UEA today who are going to come along and take some body parts off the little Ocelot, because also, as sad as this is, and as sad as it is, the animal's died and been put to sleep and it's no longer helping anyone or entertaining anyone or being there for anything in terms of a zoo's purpose, we can use these for research. We can help our understanding of these animals in the wild, in captivity, their reproduction. So we've even got a bunch of scientists who are going to come along and explain to you what work they're doing as well. Now, over the next couple of hours, you can come in, go out, you can ask us questions. We've got my wonderful team next to me on this side, and I'll be with them looking at all these skulls here. So we've got lots of different skulls on the table, and I want to look at why there are lots of different types of cats, things that look like cats, why the cats have a very particular body shape and a certain body plan. So we'll be talking about those, and you've got Jess and Catherine who will be leading the dissection today. Now, as Jess will tell you in a moment, we always have a protocol that we go through certain parts. We don't just go, I'm going to start here, and just randomly start somewhere. It'd be rubbish. And there's a temptation to do all the cool bits first, but we have to work through things in a certain order. So Jess will be going through the body of the Ocelot in a certain order here. Now you can see at the front, as I said, you can see on the different screens we have in the tent as well today. Feel free to ask us questions throughout. It will smell at some point throughout today. That's natural. The smell of an animal, this is decomposing. They do have certain smells. Certain animals smell worse. If you were here, anyone here last year for the llama? How did that smell? Bad. Expert opinion there, bad. There was no hesitation. This will not smell anywhere near as bad as the llama did last year. I think we're all very happy about that, right? Yeah. Okay. Anything to add before we start, do you think?
No, I think you really covered it all, but just to say that one of the really useful things that we get from doing these post-mortems is that it's not very often that we get to see inside these animals' bodies, because there are a limited number of them in this country, a limited number of opportunities for vets to look inside these animals' bodies, learn how they work. And the only way you can help an animal when its body goes wrong is to know how its body works normally when everything's going right. So at vet school, one of the first things that we learn, in fact, I spent 3 years learning how everything worked when everything was going right, you know, before I even started to think about how things could go wrong. So this is a learning experience for me as well. I've never dissected an ocelot before, but I will know now in the future if I'm treating ocelots about how its body works inside, and that'll be really useful for me as a vet when I'm deciding how to treat it and to understand sort of why its body is acting the way that it is. So a learning experience for you guys, a learning experience for us, and most importantly, for improving the welfare and health of these guys in the future as well.
Brilliant. So you've done lots of cats before as a vet.
Yeah.
I can imagine you work with cats, dogs, hamsters, guinea pigs.
Yeah.
Yeah?
Yeah.
Are these the same as every other cat? So I've got 2 cats at home, Newbie and Minnie. I guess they're just slightly bigger versions of the cats that I've got at home?
I mean, in some ways they are. They have lots of similar features, and they'll have lots of similar features to one another as well. But there are obviously some huge differences as well because of the size of them and because of the way that they live, the environments that they live in. These actually live in kind of similar environments, but most of the cats that we have at home came from sort of a desert environment, whereas these guys are from, well, where are they from? Do we know who?
So these are, well, yeah. Does anyone know where these are found in the wild?
Should we start with the Jaguar?
Yeah.
Although they're similar.
Yep. South America? Yeah, pretty good. What sort of habitat? Deserts and savanna? Jungle. Top answer there.
And that's why they have this kind of coat. So it doesn't look particularly jungly. You'd think, you know, maybe why wouldn't they be green or something if they're living in the middle of the jungle? But actually, the point of these spots, well, they're called rosettes actually, is to break up that outline as they're moving through the undergrowth. And it's sort of meant to be like dappled sunlight falling through the forest. You don't want to be an animal that's one bold colour because no plants are Jaguar shaped, obviously. So you want to break up that outline. And they've both, you know, got a kind of similar spotted pattern, but can anyone think of any other spotted cats? Yeah. A cheetah. So do you think a cheetah's spots are similar to this? Anyone think what a cheetah's spots are like? A leopard, yeah, different kind of spots. Does anybody know how you can tell the difference between those by looking at their spots? Leopard, jaguar, cheetah. Yeah. Yeah, so cheetahs have full black dots. Leopards have sort of circles with a hole in the middle, and jaguars have a rosette circle with a dot in the middle. So just by looking at their coats, you know that they're different. But their bodies are vastly different shapes as well. Can you remember when we did a cheetah? It looked completely different to this, didn't it?
Yep.
What were some of the main differences you can remember?
Very muscular bum and hind legs, really powerful because those running cheetahs, these adaptations are really fast sprinting animals, whereas this won't sprint very fast or very far within a forest. This is an ambush hunter. So as Jess has already said, having this lovely coloration that blends in perfectly to the surroundings. The next time you're walking in a forest or one of our Norfolk Wildlife Trust reserves, look in the undergrowth when it's light and just look at the dappling of the light on the leaves and how it all changes and looks a little bit like this. This animal wants to get as close as it can to its prey and sneak up on them without being noticed and suddenly pounce. Whereas a cheetah runs and runs and runs and tries to catch its prey before it runs out of breath. So there are differences in the way their bodies look as well. And cheetahs have a massive nose. They're just letting loads of air in to breathe as much as possible. If you're running, next time, do a little experiment for me later. Don't go very far. Try running while pinching your nose. Anyone falls over or faints, not my problem. Mums and dads. But try running holding your nose. It's really tough. And then try running, breathing heavily. If you've got even bigger nostrils and much larger nasal cavity, that's what helps the cheetahs run as fast as they can. But as we'll see with something like the jaguar, this is a massive head. This is a huge head, massive muscles, really big chest, there's so much power here, isn't there?
Yeah, and if you're thinking about dogs, for example, I'd say a cheetah is like a greyhound. Like there's nothing on it. It's really, it's got long legs. It's thin. It's really light and can run really fast. Whereas something like a jaguar is more like a staffy. You know, it's stocky. It's really so strong. Its jaws are so strong. It's not built for running those fast sprints. It's built for attacking its prey when it gets to it. And the ocelot is kind of somewhere in the middle, I would say. So it's not quite as stocky as the jaguar. Obviously, it's not as big, but it's sort of in comparison, you know, its head's not quite so big. That's because it has two methods of prey capture. It has the move and sit method. So these guys are constantly moving about their territory, but the two ways that they will catch their prey is they will either slow walk, so they'll walk really slowly, hopefully camouflaged by the light. They're hunting at night time, and they move really slowly, trying not to be seen, and then will leap on something. Or they have the move and sit, where they move to one area, they sit down, totally camouflaged, and wait for something to leap on. So they're much more sort of in the jaguar realm of chasing rather than a cheetah. And you can see that as well in their paws. So for the size of their body, these paws are really quite big. And the jaguars obviously are really enormous as well. And why do you think they might be? They're really hairy as well. You can see sort of under there. They're really padded and really hairy. And that's not to keep them warm, because the environments that they live in are pretty warm. So why do you think they have this hair here and these padded paws? Pardon? Yeah, so they're big for sort of batting them down. Yeah, and they have sharp claws as well for catching their prey. Yeah, that's one reason.
Is it like how to keep grip when
Use their claws for that, certainly, and actually that's why it's difficult for cats to come down trees as well, because their claws are sort of scythe-shaped that way, which makes it easy to get up, but then that actually makes it quite difficult because they have to pull it out when they're getting back down. So the claws definitely help for climbing trees, and actually these fifth digits, these sort of what would be our thumbs, they can sort of go around the side of tree branches as well. But there's another reason, it's to do with stealth. Exactly right, so it's to muffle the sound of their footsteps. So they're really, really padded. It sort of keeps them completely silent as they're either creeping up on their prey or doing that really small burst of an ambush hunt.
Absolutely. So what we're going to do over the next few moments is start to look at, Jess is going to start to hopefully talk us through what she's doing when she starts a post-mortem. And I'd like to know why you do post-mortems, what they can tell us, what things Jess will be looking for, but then she's going to start with a small cut down the front of the ocelot, and her and Catherine will start to look inside. All the way through, if you've got questions, stick a hand up, ask me, and I'll try and ask Jess as we're doing this. Remember, this is a live necropsy, a live dissection. This is being done not for your benefit. As nice as it is that you're here, this has to be done legally anyway, so we're using this as a real opportunity to try and engage the science with you to see what we're doing as a community and to see how we understand these incredible animals. We've also got one of our wonderful colleagues over here who's going to step in a moment, explain who he is, introduce himself, and why he's taking certain body parts off our ocelot and how it'll help understand lots of different animals in a way that we didn't before. So, Jess, what are we doing?
So you asked me there why we do post-mortems. So post-mortems are generally to find out the cause of death. Sometimes we know the cause of death and we're just going through to see if anything else was going on, to learn about the condition of that animal when it died, for our further understanding of the anatomy. But with zoo animals, it's a legal requirement that everything gets post-mortemed. And it can help to know a bit about the conditions they were in before they died as well, and if there's anything, for example, if they're living in a group, if there's anything that needs to be changed, maybe the diet that they're on is having certain effects on the organs inside their body, and we didn't know that by looking at them, but by doing a post-mortem we can say, oh, actually they were a bit fatty, or the kidneys were a bit XYZ, and maybe we should add some more fat into their diet or add some more protein. You know, we can get lots more information by looking inside these animals' bodies, perhaps about the group that they live with, not necessarily about that individual animal. So the first thing that we do is we look at the external, well, it's going to be a while because we're going to look at the external first. So what sort of things do you think you might see on the outside of the body that would give you clues to how well it was when it died, why it might have died? What do you think I might look for on the outside of the body?
Yeah. Any cuts?
Perfect, cuts, yeah. So had it hurt itself on the outside somehow? Are there any abrasions? Is there any, well, no, I won't say that because there's another answer.
Has it been in a fight, maybe?
Has it been in a fight? Yes. Is there anything in its cage that might have injured it that we might need to remove? Yeah, fights is a really good one to work out the dynamics of the group.
Tooth decay. Wow.
Excellent, yeah, so we can...
Hang on. Are you a vet by any chance? you might be, just a very young-looking vet there. But tooth decay is a really good one, isn't it, Jess?
Yeah, we can tell a lot about health from the teeth. We can tell, and sometimes we don't know how old these animals are, so that can be, teeth can be a really good indicator.
While we're there, on this Jaguar, you can see in the canine here, can we just turn this camera to see this one, is that possible? So you can see this lovely lower right canine here has broken off at some point. So you can see these big canines at the side here, massive canines as you'd expect. This one's broken. Now you wouldn't often see that in the wild to this level, would you? You see more tooth decay, more breaks, more fractures in
But actually
animals.
in these two individuals, I would say that is because of their age as well.
Yeah.
So these two individuals lived a lot longer than they probably would have done in the wild. They were both very, very old individuals when they died. They probably never would have reached those ages in the wild because, you know, there are lots more threats, there are other predators. Once they start to sort of get old and arthritic, they're going to be outcompeted by other animals that are younger and healthier. So, and
also you can, sorry Jess, but you can see a little bit of plaque build up on some of these big teeth here as well. You wouldn't usually see that in wild cats. And as you say, they're living longer.
Yeah.
The longer we live, grannies, granddads sometimes don't have their normal, their real teeth, they have false teeth, and we can't give tigers and lions and jaguars false teeth, but we see their teeth decaying and changing over time. So this is, as Jess said, just a sign of an older cat in captivity, and also their diets aren't always quite the same.
Yeah, and also their keepers aren't regularly volunteering to go in and clean their teeth.
Anyone volunteering to go and brush some jaguar's teeth in the future? A couple of you, yeah, brilliant, great.
So, you know, with pets, we do encourage to clean their teeth, but with an animal like this, you don't want to be going inside and getting anywhere near their mouth. So, yeah, but it's completely normal. And actually, this one's teeth, I'd say, are pretty good. I mean, still extremely sharp, actually.
So Jess has said something that sounds obvious, that it's extremely sharp. Anyone got a dog at home? Yeah? Your dog's teeth aren't especially sharp, are they, really? Because
actually, that ones are not particularly sharp.
The side ones are.
Those ones are, but the canines even on this one are sharp.
So they're quite nippy, aren't they?
Yeah.
They're quite sharp, little canines. I mean, this is bigger, but these are much stockier proportionally.
That's about pressure.
Yeah, this is grabbing, this is choking, this is killing, this is breaking skulls. That
and it
one's
has
not,
that huge head for the musculature to hold its jaw shut.
Yeah.
Whereas this one needs to be sort of a bit sharper.
And it'll be good at some point when we're not doing this quite as intently as we are now. Have a look at these skulls here, and even though most of these, or many of these, are cats, you can see that two very different cat species here look very, very different from one another just on their teeth alone. So I want you to start thinking, why do different cats have such different tooth morphology as well, and what does that allow them to do, what does it not allow them to do? But one of the big differences between the cat groups are the shape, the size, the sharpness, and differences within their teeth morphology. It's quite cool, isn't it?
And cats actually don't have that many teeth. So a dog, for example, has 42 teeth, whereas a cat only has 30. They have much short. I mean, you can see that just by looking at a cat's head compared to a dog, a wolf-like dog, has a long snout and much more space for teeth. Now, cats' faces are much reduced, and they. Well, do you want to talk about carnassials? You like to talk about them, don't you?
It's one of my favourite things in biology. Oh, I get really excited about this. So if you put your finger, I'm not going to do it because I've been touching this jaguar, put your finger in the back of your mouth and bite down very gently. Don't injure your own fingers. I'm not going to do health and safety with that. You should feel a lot of pressure because you've got big teeth crunching down on a small area. I stick it in there. If you stick your finger in a lion's mouth, A, you're really stupid, but B, you wouldn't feel that nice chunky pressure. You would go, oh, where's my finger gone? Because it would slice through them. So as Jess mentioned, they've lost a lot of their teeth. So dogs have got 42 teeth, cats have got 30 teeth. How many teeth have we got? As 30, how many?
30.
30. How many teeth have humans got as adults? 32, yes. So we've got more in terms of, more similar to cats, I guess. But the differences are, they've got these big canines, but also at the side, these things called carnassials. Now, these are like big shearing teeth that we see in kitchen knives that just shear against one another. And because they can't cut their food up, so hopefully, I'm guessing most of you use knives and forks or spoons or chopsticks or different tools to eat most of your food. They can't do this. They're using their paws to take things down, but they're not cutting things up. They're using their teeth and their tongues to process their food, to make them into smaller bite-sized chunks. And a big way of doing that are these big, sharp teeth at the side. Now, do you want to put your finger inside that's mouth very gently? No. Do you want to put your fingers in there? At the side where it's really sharp? There's nothing wrong with that bit. Now stick them in the side. Now imagine that going down there. What's going to happen to your finger?
Get cut off.
It's going to get cut off, isn't it? Yeah. Very brave there. I like the amount you trusted me. Thank you. So Jess already is going to look at these teeth and start to look for fractures, start to look for any sort of wear here. But these are a really important part of the cat's evolution to have these different teeth. Yes?
How hard can a jaguar bite or a leopard bite compared to a human?
How hard can a leopard bite compare to a human? It's in terms of bite force, I don't know the exact numbers, you'll probably have a bigger bite force than a leopard, purely because the leopard's relying on physics. This is more of a Brian Cox thing. Brian's not here right now, but I can try and say like Brian, I've finished looking at planets, I'm going to talk about the physics. What Brian would say, I assume, is you've got a huge amount of force going into a very small area. So it doesn't need a massive set of muscles. So leopard musculature isn't especially advanced or built up like you see in this Jaguar here, but it's relying on these very small points of pressure to just penetrate, to puncture and kill. This is different, however. This will have a much greater bite force than what we do. So we're talking in terms of pounds per square inch. We have, I think, something like, it's like a couple of hundred pounds per square inch in terms of bite force. You're talking, about 3,000 psi on something like this, so it's much, much larger. So it's a really huge amount of force in quite a small area as well, which is why it can bite through the backs of skulls of things like capybaras. So it's beautifully adapted to fit right at the back of this giant guinea pig. So it fits there beautifully, first of all, as you can see. That's the kill bite, but there's a huge amount of force going into the back of that skull to destroy that, to disable that animal there. Okay, Jess, sorry, I'm waffling.
So I'm just continuing looking at the external anatomy. Just moved down to the claws now. So not so much it's just habit that I'm doing this, because if a domestic cat came in, for example, and it was dead or injured even, sometimes they come in and we don't know why. One of the things we look at is claws, because if a cat has been in a car accident, then its claws, it's often tried to grab the road and its claws are usually shredded. And you know, it's really good practice to just look at all parts of the animal. And again, this cat's, apart from this one fifth digit, which does look a bit broken, but I think that's just incidental. Its claws looking great. So it was in teeth and claws were in fighting fit condition, even though it was 15. It was still.
And that's something we see in all the cats, this ability to have these wonderful claws that can move out and back in. Now there's an urban myth that one cat can't retract its claws. Which cat can't retract its claws? Cheetah. Now we did a cheetah dissection, didn't we?
We got told off.
And we got told off. We did it for the Radio 4, Wild Inside, Ben and Jess. And we were about to go, well, the cheetah has claws that cannot be retracted. And what do the claws do? Retracted.
Well, no, actually, we're not supposed to say that. Don't you remember? You're supposed to say
know.
protractile.
They protracted. So, I mean, we think we're quite nerdy and pedantic. We met a very nerdy and pedantic friend of ours.
Awesome guy.
Yeah, he was lovely. But Cheetah claws do move in and out. So we've always taught, I taught my students, I was taught as a student that they're just like running spikes and they don't move. And we found this wonderful cheetah and you press its paws and the claws go in, the claws go out, the claws go in.
And so what he told us was that cats hold their claws in most of the time. So that's their natural position. So rather than us saying retractile, we should be saying protractile because they're only pushing them out when they need them, rather than
we can show that really nicely. If I just manage to turn this here. Which one's best for showing? Ah, there we are. So you can see this lovely little danger mitten. Nice and cute. High fives or high fours all round, no problem. And then, oh, food's turning up. And that thing comes out. So that's its natural position, as Jess said. So it's not retractable. They're not there and they're pulled back. It's the opposite. So in their natural position, the tendons, the ligaments, all keep them high in the paw, the front or the back. And when it needs to, it'll push these big claws out. And you can see by pressing here. And if you've got a cat at home and your cat really is very friendly, I wouldn't do this with mine, you can press their paws and make their claws come out. Obviously don't do it if they don't want to, but and also don't try it on a live Jaguar. But as you can see, these massive claws come shooting out the moment you start to press on the pads here. And we've got this massive one at the side somewhere as well. If I can just push that one. That's the one that's doing the danger work. That's the kill. That's the one that's taking them down, hooking the legs in here. So you can see these massive claws. So Jess has had a good look at the claws on our little Ocelot here. But this, again, is another lovely adaptation that we see in all the cats, isn't it?
Just ripped my glove.
Did you?
Claws are too sharp. They ripped my glove. Right. Good. Where were we? So the coat. So anyone think of anything else you might want to look at on the outside? So we said whether it would have any wounds. Well, I'll just tell you. Yeah. The colour of its fur. So the colour of its fur probably isn't likely to change, but what is likely to change is maybe some bald patches, so some areas where there isn't fur, where there should be fur, and also the condition of the skin, so whether it's flaky, scurfy, oily. But actually, all of the external condition of this cat looks pretty good. We also look at muscle cover, so whether they were sort of wasting at all, and actually this has got some wastage over the back, which is really, really normal for an older cat. You'd expect some arthritis, probably not moving about as much as it used to. We know this was a very old individual, so I wouldn't say that that's a cause of death, but it's just something expected.
Yep.
And do you think for parasites?
Parasites. We look for parasites.
Yes, and actually, yes, we would look. So most parasites on the external body would leave when the animal died, because obviously they mostly feed on blood, and so the blood's not flowing anymore, so they've got nothing to eat, so they're going to go away. So quite often when we get a dead animal in for a post-mortem, we open the box and we get like an army of fleas or mites or whatever sort of crawling out of the box, which is always really, really pleasant. So we often do see inside the body, sort of, if we've got worms and things like that, they've got nowhere to go, so we often find those. But on the external, the mites, the ticks, the fleas, the ones that can get away, they're a bit like rats on a sinking ship, is that the...
Yeah, and this is something we'd see more in a wild animal than we would usually a domestic, like a pet at home, or a zoo animal as well. We can treat these animals so you wouldn't expect to see a huge amount of parasites in an animal that's been kept really well, that's been maintained. Although we did a porpoise dissection last year, and it had worms next to its worms next to its worms. Even in the blowhole, the worms were coming out. It was like Bella Italia from hell.
Yeah.
Sorry, everyone. But it was not fun.
It was amazing.
It was amazing.
And I love parasites.
One of us loves parasites.
Was even a bit...
One of us is normal. Yes. Could it have broken bones?
It could have broken bones, and so you would be checking for all of the bones that you can feel as well.
How do you check? You haven't got X-ray vision, so how do you know what those bones are doing right now?
So I mean, you can feel them through but you don't know 100%.
So but you're moving. I see you're moving the leg in a nice way.
Yes.
So you're looking for the way it moves and doesn't move.
Yeah, certainly. And you don't want it sort of bending anywhere it shouldn't have a bend. Or So
any broken bones you think so far?
No.
No. Cool.
And we we have this animal's history before it died, and I would expect some kind of pain or some limping or, you know, a history of some kind of problem like that. So sometimes we get clues before you do the post-mortem. You know, it often comes with sort of a bit of information. It's clinical history, it's called. So the things the keepers have been concerned about, the drugs that it's been on, things like that. In this case, it was an old animal that just wasn't doing so well, which, you know, is quite a common way that we see things. But yeah, broken bones, no. Ears, I'm going to move on to now. It's because, as well, you were talking about parasites, and I think this cat probably did have some parasites in its ears. Really, really common for cats to get ear mites. You know, very often we don't even treat them because they sort of manage with them, and unless they're really, really super itchy, you know, they can do fine. But there's a lot of wax in here. Could just be normal earwax, but it's quite dark. So there may have been a few ear mites in there, but that is definitely nothing to be concerned about. Cats' ears are amazing. They are really muscular, and they're able to move really in quite a wide area to be able to, this bit, the pinner here, to funnel sound down. And they do rely on sound a lot when they're hunting. Dogs rely a lot more on scent, but cats rely a lot on sound. So they've got excellent ears and eyes. Now, this animal was frozen. Very often, if we can't do the post-mortem immediately when it died, which I'd say with zoo animals is 90% of the time, they are frozen before we do the post-mortem. And unfortunately, the freezing process sort of distorts the eyes quite a lot and makes it quite difficult to see them. But obviously, I'd be able to see if there was any physical damage to the surface of the eye or to the area around the eye. It looks fine. But yeah, in a non-frozen dissection, you might get the eyes out and have a look at the inside, but there's not really any point in a frozen one. You want to talk about tapetum?
Yeah, so if anyone's seen a cat at home and you shine your cat, don't shine your cat, you shine your torch to look for your cat. Mine are often in the garden, they have a quick wee at night, they'll come back in very quickly, so I don't want my cats to be outside too long. And you see these two little green lights flashing back at you. That's not because your cat has special torches in their eyes, because they've got a mirror-like substance or a mirror-like piece of anatomy at the back of their eyes. This is called the tapetum lucidum, and it's a reflective lens that sits at the back of the eye to trap as much available light as possible, and then bouncing that around the eye to allow it to see almost perfectly in the dark, which is why your cat shouldn't bump into things at night in the same way that you do. And so if you shine your light, you can see your tapetum lucidum. Now, as Jess says, sometimes we can take the eye out and have a look at this. We might see if we can. It probably won't work. But occasionally, we did a cheetah up at a different science festival. Apparently, there are others out there a few years ago. And we did see the tapetum lucidum in a cheetah. But this is a much smaller animal. So Jess has also got our little ocelot head on here. And we've got these lovely long whiskers. And what I love is that the whiskers on your ocelot are almost, well, probably longer than the whiskers on my Jaguar here as well. Why is that? Why do you reckon? Any idea? I mean, I think I've got an idea, but I don't really know.
What's your idea first?
So, nicely done. So my idea is that these things are hunting much larger prey usually and can see or hear, or it's much easier to go through the undergrowth and go for a caiman or to attack a capybara. These will be going for much smaller animals often. They'll take small capybara, but because they're lower in the vegetation, they'll be going through the vegetation and sneaking through, and they need to be much more aware of their initial surroundings. And like all these cats and dogs and other animals with these sensory whiskers, they're feeling the world around them from the point of view of these wonderful, wiry, what we call vibrissae, which are these hairs here. The Jaguar is not using them in the same way that the little ocelot is. So more importantly, this one's using its whiskers much more frequently and much more to survive and avoid predators itself and to catch prey than something like our Jaguar here. So they're both using sensory whiskers, but much more so for our little ocelot next to us.
I agree.
Yeah? Oh,
yeah.
Thank you.
So last bit, just checking the paw pads. They all look fine. So as we said before, very hairy, very padded for a quiet approach. They're also shock absorbers, so these guys climb trees, and when they're leaping down, we want something to absorb the pressure of the landing. These guys also sweat, so I don't know, have any of you got pet cats? Yeah? When you've taken them to the vets, have you ever noticed what gets left on the table when they walk around? They left paw prints. So this is where they sweat. They're covered in hair all over their body. This is their main point of sweating. So paw pads, really, really important. You don't want any damage to those. Also, they are super, super sensitive. So those of you who have pet cats probably know they don't like to be touched on them very much. And that's because they're so, so super sensitive. So any small, quite often cats come in absolutely limping for the tiniest, tiniest little wound on their foot. And that's because they're just so super sensitive. But these look absolutely fine. And last thing to look at in the mouth, you mentioned it briefly when we were talking about teeth.
Yep.
Cat tongues. We love cat tongues.
We love cat tongues. We do love cat tongues.
So if any of you have been licked by a cat, you might have been licked by your pet cat, what does it feel like? Rough. It's not slobbery like a dog, is it? It's actually almost quite painful because it's so rough. And that's because they have these amazing keratin spikes, which are huge on, sorry.
Keratin. Where have we heard that word before? Where have who's got a spiky tongue here? There's always one. You shouldn't have it. You need to go see a doctor if you've got a spiky tongue. You might be part cat. You shouldn't have a spiky tongue, but you've all got keratin in and on your bodies. Where might you have keratin? Yes. Fingernails? Anywhere else? Hair? Yes. So your hair is made of keratin, your fingernails are made of keratin, and we're seeing the same thing on these tongues here. So as Jess said, when your cat at home gives you a lick, that's the keratin. And you can hear... If you put your microphone next to it, does that work?
Where is my microphone?
On your throat.
Can you hear that?
No.
Try it again. We get a photo this time.
Yeah, do that one. Oh, do it on the Jaguar.
Just like sandpaper. Yeah, it really is like sandpaper. And you can see from the photo here, from the screen here, much, much larger as you'd expect on a much larger cat. But they're both seeing these very large keratinized spikes on their tongue. And they're all facing in one direction, aren't they? Why would they all be facing in one direction? Why would you want a spiky tongue full of spines with backward-facing, sharp little extra teeth almost? Yeah. Absolutely, they are taking flesh off your bones. So when Tiddles at home is giving you a little lick later, think, oh, that's cute. She's actually thinking of stripping the flesh off your bones to eat you.
There are two other
functions.
Cats are
monsters.
Two other functions. Does anyone get any other ideas what the spikes on the tongue might be for? Yeah, so they're pointing in the right direction for the food to go down the throat. Very good. They're in the top of the roof of the mouth as well, yeah?
They use them to clean parts of their mouth as well. They clean themselves.
To clean their mouth? Maybe.
Yeah, possibly.
Sure they do use them for cleaning. Which bit of their body might they use them for cleaning? Their paws, yeah, and
Where else? You can say paws? Yeah, everywhere. Just all parts.
Everywhere, yeah. It's like a comb. So it's like the bristles on a comb. So cats are fastidious about keeping themselves clean. So they use their tongue just like a comb to brush their hair. And it also holds onto saliva. So that spreads the saliva all over their body, which can have a function in cooling, keeping them clean. They're just really useful little objects, these spiky tongues. I think we've missed out there.
I'd like a spiky tongue.
Yeah.
It would. I don't brush my hair very often, as you can probably tell, but it would be much easier if I could spiky tongue myself.
So I think
Are we done next? Because we might be a good time to introduce our resident scientist.
Oh, yes, sorry. Yes.
So as I said earlier, we've got a guest visiting us today who's going to introduce himself, explain why he's here, what he's doing, and why he wants certain bits of our Ocelot. Oh, in a second.
Hello? Hello? Yeah. Hi, my name's Johnny. I'm a PhD student at the University of East Anglia. And essentially, I'm interested in reproduction. So how do we reproduce? How does it affect individuals in general? And I'm part of a project that's looking at how reproduction differs across all the species. So we have a lot of fish, we have a lot of humans, and so this is just a great opportunity to see what it's like in big cats.
So what sort of thing are you looking at today? What do you need?
And
remember, there are children here.
Yeah. So I will need the Ocelot's testicles.
Just the testicles.
Just the testicles, yes.
So the boy bits.
The boy bits.
We need the boy bits off, and there are some big boy bits on this little cat here. You can probably see. So if anyone's delicate sensitivities,
Should I just chop them out now?
do you just want to chop them off? We're going in here hard and heavy today, folks. So the really nice thing about, and this seems, and we're all having a bit of a laugh, but actually the really nice thing is here, is this animal isn't just being used for the post-mortem, it isn't just being used for the opportunity of a science festival. You've got, as Johnny said here, this is a big team of people who are working across different animals from different groups, from different taxonomic parts of the animal kingdom, from fish to mammals and everything in between. And it allows his team to really explore how different animals reproduce, why different animals do things in certain ways. And will there be any conservation benefits potentially for something like this, do you think?
Well, yes. So if we understand how they reproduce, we can then focus any conservation efforts based on the results. So if we understand that, for example, birds reproduce better in early age, we would then concentrate on getting birds younger to reproduce faster.
And that's what, as Jess was saying earlier, this is a nice opportunity. So for Jess, she's learning today about the anatomy of an ocelot, if it comes up again. This has applied conservation benefits. Johnny's here with the rest of his team who will be looking at these bits and bobs of different animals from all over the animal kingdom to see if there are ways to understand their evolution, but equally as importantly, ways to understand how we can help them in conservation, how we can help them in captive management, and all different things here as well. So, Jess, you have, something's popped out.
Sorry. So the reproductive organs of the cat sit really high up, right under the anus, not like in dogs where they have quite pendulous testicles and the penis is more down here. In the cat, it's all right up here. They have a really, really tiny penis, which is also barbed. As you can see there.
I know that at least 2 or 3 parents are going, I thought this was a family thing. They told me it was great for the kids.
Science.
It is science, but I can still guarantee there's at least one parent going, this is not how I thought I was going to spend my Saturday morning. 11:43, extending an ocelot willy. Come to Norwich Science Festival.
So there we go. Can you see that? Is it on the screen?
Yep,
Yeah.
It is on the screen. If you all want a closer look at the ocelot penis, it is there.
And
now,
so it's
Really barbed.
you said
it's really barbed, yeah.
So why are they spiky? I get why the tongue's spiky now.
Yeah.
Why would the willy, the penis, be spiky?
So I think my cat penis knowledge is a little bit out of date, but from memory, I think it's because they're induced ovulators, which means unlike other mammals, like us, for example, we have a reproductive cycle where we have a fertile window every month. And so if we mate, if we
Want to make a baby?
want to make a baby at a certain time of a month, we have a fertile period. These cats are generally solitary. They don't come together very often. And so when they do, and they come together and mate, you know, it's not a monthly occurrence, for example. And they want to make sure that they are fertilized when they mate. So these barbs, from what I remember, induce ovulation. So they release some sort of hormone surge, which causes the female to reduce an egg at that moment. So it's actually the mating that makes her become fertile, rather than a monthly cycle of hormones, which makes her have a fertile window. Do you know about that?
No, I didn't.
No?
I thought it was more.
I think that's right.
It helps stop other males mating with the female because they kind of get a bit stuck.
It does that as well. It does both. So it's multifunctional. So it does that. It induces
Okay.
ovulation. It also, as Johnny said, it helps competition. If another male has met that female,
Scraped it out.
it'll help pull, some of the previous sperm out. I think we should just carry on, to be honest. I'm way too deep here, so to speak.
Oh, Ben.
So, we need to be very careful and store this afterwards, don't we? We can't have them. Obviously, we're in the environment here, and the longer we leave any tissue, any soft tissue like muscle or lung or the testicles, the bits here, the boy bits, you can't do as much with them. So you need to prepare these in a certain way, get them off, take them and store them. How do we store these?
So we are going to essentially freeze them again.
Okay.
So keep them on ice and then put them in a freezer.
Okay, cool.
Because that kind of keeps everything that we need about the testicles in optimum condition.
It's really friable, I'm afraid.
That's all right. So if you become scientists, kids, this is the sort of thing you can do. Yes?
There it is. You can see the epididymis.
Yeah.
Any evolutionary adaptation to avoid being scraped off?
Maybe. It's yeah. In terms of a lot of the question there is, I guess, there's lots of competition between individuals in terms of who's bigger, who's more aggressive, but also reproductively. There's still competition there. Who makes the most sperm, is the most active, and if you can remove some others as well. So it's a different level of competition. So we think there's probably an element of removing previous as well. What you're noticing today, everyone, is that this isn't a fast process. We could have done this in about 25 minutes. Welcome, everyone. Here's a cat. Right, see you later. Bye. But as you're seeing with what Jess is doing, she's thorough. She's going through everything stage by stage, because what she could notice is one tiny little thing, one tiny discoloration or one tiny twist of an organ or a tube somewhere or a vessel that could have been problematic for the animal in front of her. So this is a slow process. And regardless, whatever sort of scientist you might become or want to become, whether you're a physicist or a biologist or a vet, we are all very thorough, we are slow, we are methodical, and I think that's what we're seeing today, and it's really nice that you're seeing real science. And very often you might see it on TV and it's, I thought of an idea, and then I went to space. There might be 20 years between those two things happening, and even the stuff that we do, there might be weeks or months between having an idea, sorting a solution out, and actually making that happen. You can see this in real time. Sometimes science is quite a slow process. And even something as simple as removing these bits to give to Johnny that he can do his research sounds like it's really quick, it's really simple. But as you can see, it's taking its time here. We talked earlier about this big Jaguar and, oh, sorry. Go on then. Johnny's got more stuff to talk about here.
So essentially, what we can see here is the actual testicle. So that's the bit that produces all the information needed to reproduce from a male. So the globe at the top, essentially, is what produces the sperm needed for reproduction, and then it travels down through the tube. I don't know if you can see it, but essentially downwards, and then that comes out through the penis. So I'm only interested in the testicle part, because I'm only interested in the actual organ that produces the sperm. So we'll probably just cut underneath.
Anyone any questions for Johnny while we're here? Because he's not going to be here for much longer. Any questions on the research he's doing, or yeah?
Are you only researching the reproduction of higher
You can put it down now.
Just higher animals, or what are you looking at?
Essentially anything. So we are really interested, because we have a lot of very specific pictures of how animals reproduce, but it's quite difficult to get an overall aspect. So we're doing horses, cows, all of more farm animals, but then we're also doing fish, frogs. We've got a lot of amphibians. We've got axolotl as well.
What about more, I guess, like something like the Darian, jellyfish?
So, well, do you think, so one of the reasons, why do you think we might not be able to use those organisms while looking at reproduction necessarily as easily? No, so essentially all the limit of what we can use for organisms are ones that produce sperm. And essentially some of the lower organisms don't produce sperm. They have very unique and very complicated life cycles. So although that would be really interesting, it makes it difficult to compare. So we're more focusing on more of those.
Yep.
How big is Johnny's collection of male reproductive organs?
Get out.
Shockingly big.
So that's a good question. How many? What sort of sample size are you looking at? I mean, is this number 10, or do you hope to have 10,000, or how many do we need to do good research? Really?
Well, so essentially, it depends what aspect you're looking at, but you'd want at least 2 individuals per species.
Yep.
One young, one old, so you can do a comparison. But for example, some species are easier to come by than others, so we have around 60 sheep testicles. So that's a huge sample size. We have quite a lot of fish because they're quite easy. But some of the rarer ones, so we want to get, for example, a platypus, but we'll only get 2 or 1 minimum.
Yeah.
What are you going to do with it? Are you going to dissect it to try and understand how it works, or are you experimenting?
So what we're going to do is that we're going to essentially take some of the tissues from right in the middle, because those are specifically what I'm interested in, or what my group is interested in, because they're the bits that produce the sperm. So we're going to isolate them and then essentially understand what kind of mutations there are within those cells compared to other species and how they differ.
Yes.
Do you do reptiles?
Reptiles. I would love to do reptiles. We don't have any yet, but hopefully at some point that would be really cool. Thank you, Ben.
So when we castrate a cat at the vets, this is exactly what we do. Make an incision on the outside of the scrotum, and then remove the testicles from inside. And then, I mean, you'd either tie it with suture material, or you can just tie a knot on itself, because obviously this is a massive blood vessel here. So you don't want the blood to come out of the body, so you need to end the blood supply. You need to stop the blood from leaking out. So you either tie a suture around there, or tie it on itself, so knot it on itself, and then just cut them off. That's how you castrate a cat. Simple as that.
There's a skill you didn't think you were going to learn today, hey folks? Please still send your cats to the vets if you need to castrate one. That's not what we're endorsing today.
I don't know why I am actually castrating it. I don't need to do
I don't know why you're doing
it's
it
just
either.
habit. Right. Oh, sorry.
Scientific. Okay. Very scientific.
Today's castration is sponsored by Tesco's.
So there is a sciencey pot over there. Yes, so I've got some beetles and fruit flies. So these are really small, so you can imagine their reproductive organs are really, really tiny, so you need a microscope to kind of tweeze them apart. So what's the focus of my research? So essentially, we just want to understand what the general picture is. Because as I mentioned earlier, everything's very specific. So we know how humans are, and we know how a lot of farm animals are, but we don't really understand how it differs across species. So we're just trying to understand how fertility differs amongst all these species and across age.
No, we didn't have any. I think it was castrated.
Whales? No whales. But again, another thing maybe Ben will be able to help us on.
We wouldn't have I didn't take them out. I don't remember taking them out.
So we're looking more overall at the genetic landscape. So it's like mutation load across in general. Because one of the problems of why no one's really done this before is because it's quite complicated to do specific comparisons. Because as you can imagine, you might be able to compare the Ocelot and the Jaguar, but you wouldn't really necessarily be able to compare a dolphin and an Ocelot. So if you do more of a vague comparison across all of them, you can then infer more about what the differences in mutations are.
Just a quick reminder for those who've turned up who weren't here at the start. We are here today, so you've got Dr. Jess French, friend and colleague and veterinary surgeon over there, Catherine, Johnny, Emily and Ellen, and myself, Ben. We are all scientists and academics and biologists and vets and students and a whole different range of us here. The animals we have in front of us today, we've got a Jaguar here, this big beautiful Jaguar boy, and this little Ocelot. male as well. Both are from British zoos. Neither were harmed or killed for the work that we are doing today. Both were old animals. Both were sadly put to sleep because a welfare decision was made for both of them that they weren't as happy as they could be anymore. And often we see in zoo animals, especially because they're not dying of fighting or they're not dying of old age in the same way they might in the wild, they're living a lot longer than they typically would as wild counterparts. And very often the decision is made to put them to sleep for ethical reasons, for welfare reasons. So these animals have not been put to sleep for your benefit today. The nice thing is we have to do this anyway. This is a legal requirement that Jess is asked to do to make sure that we know why this ocelot was unhealthy, why it was sick, and potentially Jess can find a cause of death. And as we've seen with our wonderful friend and colleague over there, Johnny, there are often benefits that we can do for research. And as Jess was mentioning earlier, there are conservation benefits as well. If we can manage these animals better in captivity and potentially in the wild, it's great for everyone. There is a big debate around zoos, whether you like zoos, whether you don't like zoos. There's a time and a place for that, I guess, and we're happy to talk about that. Regardless, as long as zoos are around, there are still benefits to the animals that we have within these zoos. And if we can use them in a wider context with vets, with geneticists, with biologists, with students, and also with the public, then in my mind at least, these animals can benefit their wild counterparts and others in captivity as well. Now the Jaguar has already had a post-mortem, so we've got him here today for comparative benefits. We can see these huge claws that we were talking about earlier. We can see this massive musculature around the back of the head, these huge canines and carnassials, whereas Jess is now about to start the midline incision down the ocelot. So what are you doing, Jess?
So the one thing I wanted to mention here was, so we looked at the whole coat and I said there might be some bald patches. So there's a patch here. What do you notice about the shape of it first? It's a square. It's a square with very sharp edges. So do you think that looks like a clump of hair that's naturally fallen out? No? What do you think happened? Might have been given medicine. So first of all, this has definitely been made by a vet or a vet nurse, I imagine, with a pair of clippers. That's a very classic shape that you'd get from clippers. I would say probably from that position it wouldn't have been given medicine there. There's something else we do in this particular position on cats. So it's right on the neck over, well, yeah, what do you think it might be? It could be where they put it to sleep, yeah. Yeah, so it could be where they put it to sleep, and it could also be that's a very normal position for us to do a blood test. So you've got a big vein under there that we can easily take blood from. So that is not something that's going to inform my post-mortem, but just interesting to know. And now, yes, as Ben said, we're going to start the midline incision. So that's an incision that goes all the way along the body. Today I'm going to try and do it slightly differently in that we're trying to preserve the skeleton for teaching purposes. So normally you'd sort of cut the skeleton and splay it open, but I'm going to kind of try and get the organs out a bit more so we don't damage the bones. But still I'm going to start with this long incision. I'll probably do down to the muscle layer, peel the skin back a bit so we can see some of the muscles. And then the next bit we'll go into will probably be the abdominal cavity, and then we'll do the thorax.
Yes.
You know the bones?
Yeah.
Will you give them to the museum tomorrow?
The bones, what do we do them? So my plan is, so a lot of my research used to be looking at bones of different animals and see how they change over time and evolution. So what causes a new species? This is the sort of question I was talking about with my students this week. We don't really know what a species is. We think we know what a species is. I'm very confident saying these two are different species. But when we start to get to the nitty-gritty of what actually defines a species, there's still no 100% agreed definition that everyone agrees on. So by looking at what causes speciation, what helps different species evolve, the more we can look at different things, whether it's genetics, like Johnny was doing, whether it's the anatomy, whether it's the bones and the shape of the bones and the changes, we can all benefit. So these will be the bones will be staying at my university in our lab. And we'll be using them for we can fire lasers at them and look at tiny differences that are 1/100 of a millimeter difference between one bone and the next one and the next one, to look for these differences between our Jaguar in the zoo, in captivity, in the wild, old ones, young ones, boys, girls. We can use all this different type of research, and it's good for teaching as well. Jess can look at them so what sort of diseases do the bones get maybe? Do they have different? So we know that some things like elephants and some of the great apes like chimps and gorillas don't always do very well in zoos in the past, so they get bad fingers and toes, so we can look at those sorts of things as well. So there's lots of things we can look at with the bones, but these won't be going to the museum, these will be going to our laboratory, and we will share them. If people want to study them and want to look at them and work with them, then we'll make that open for everyone. Yes?
Species. Are these like any specific subspecies of Jaguar or Ocelot? Do you even have subspecies?
I don't think the Ocelot has subspecies at all. It's just one species, isn't it, the Ocelot? I don't think there are regional There are region morphological differences in some of the jags, but not enough to subspeciate. So the question there was, are there subspecies? So you get your different levels of classification. So you might have these apples are all different to these oranges, but then what sort of apples? Are they Granny Smiths or Golden Delicious? That might be what we call subspecies. So it's a different way of classifying different animals and plants and fungi and other organisms. Sometimes you get different variations across different areas. Sometimes you'll say they're different species or different subspecies or different regional morphs. It depends where you are in the world, and it also depends which group of animals you're looking at. We might classify birds slightly differently to what we do mammals. There's often we think science is right or wrong, or black or white, or the perfect or rubbish. There's lots of variation within how we look at science sometimes, which is why it's so important. And obviously, I know where you're from, I know what you're, which is why we're so important and so happy that we are always so open with this sort of science to really have that discussion to see how we tell whether this is a species or a subspecies. Does it really matter? If we're trying to save all the jaguars, does it really matter that we've separated them into regional morphs or different subspecies, for example? So I work with chimpanzees a lot. There's one species of chimp, yet there are at least 4 different subspecies of chimp. But they can all breed with each other and make successful babies. Well, I used to work in Uganda, and they look completely different to the ones that I now work with in Liberia, for example. And yet they can all breed together. And does it really matter if we save one subspecies, all the subspecies, or we've got to think about those sorts of things in conservation as well? Yes, what's your question?
Will you shave the ocelot?
Will we shave the ocelot? No.
So if I was opening this animal to do an operation, like say I was going to spay it, or it came into the vet's, and this would be a very similar incision along, you'll always do it along the midline here, if you're going into the abdominal cavity. I would 100% shave the area. Why do you think I would do that? Stop the blood getting on the fur, yeah. Owners don't like their pets to go home covered in fur, yeah. Blood.
So you can see where you're cutting?
See where you're cutting, perfect, yes. So actually this is much more difficult, and I have nicked the muscle a couple of times because I can't see what I'm doing. Whereas if I've got a perfect skin layer, I can see exactly where I'm cutting. That's much more important on a live animal where you don't want to accidentally cut something that you shouldn't. It's not really going to make any difference if I do it on this one. Although I still want to keep the organs intact because it's good practice, and it's easier to look at them. It's not a life or death situation. Another reason, so to see where I'm going, yeah, to keep the whole thing clean, yeah. To see if there's any diseases on the skin, good, so you'll get a closer look at the skin surface. Also, what don't you want to get into the wound, do you think? What don't you want inside the body? Yeah? Hair. You don't want any of that hair from the outside going in because your body's going to say, that's a foreign object, and it's going to react really badly to that. You're going to get maybe abscesses forming inside, and you might also introduce infections, yes. You might introduce bacteria, yeast, things like that, things that definitely shouldn't be inside the body. So if you're doing an operation, you would shave a large area around where the incision would be, and then you give it a really good clean with some sort of antiseptic scrub to make sure you've got rid of all of the hair and all of the possible infection there to make it as clean as possible, because you're going into an environment which is not normally open to the outside. So you don't want anything getting inside there because it won't be able to cope, and it'll mean there'll be infections and lots of post-surgical complications.
What's your question?
So obviously humans, so essentially different species, Neanderthals, etc. Of course, modern day humans, sapiens, the average person has 2 to 3% of Neanderthal DNA in there. How blurred were the lines between kind of the past species of human and the current day?
That's a really good question. So we've gone off track from cats a little bit, which I like this, but back to the species question. And your question there was, humans, most of us will have... Hang on, I'll ask you. Who here is a human? There are some people who've not put their hands up. I'm slightly confused by that. Okay. Who here is just human? Who's here is only human being? Only Homo sapiens, nothing else inside you? Few of you think you might be? No. Oh, no, no, no. You, each of us, I think everyone in here, yep, pretty much everyone in here, is at least 2, 3, maybe even 4 species all mixed together. You are not just one species. You are the wonderful culmination of some weird dating things that went slightly quirky a few 100,000 years ago or more recently. We are mostly Homo sapien. However, most of us in this room, looking around, most of us will have between 2 and as much as 6% Neanderthal DNA in us. Don't look around for someone who you think might be slightly more Neanderthal. All right, it doesn't work like that. I can just see now, my dad looks more like a Neanderthal. Doesn't work like that. You'll also have Denisovans inside you as well, which is a Siberian extinct human, about 1%. And we even have about 1% ghost species. Ooh. Doesn't mean that. It just means we don't know what that 1% is from so far. It might be Homo erectus, which is too old now to look at the DNA. We can't extract that DNA. Or there might be another species that we haven't discovered yet. So everyone in this room is not purely Homo sapien. I love that. So this whole idea of species again, what even is a species? Or even this one thing is like some weird mishmash of a recipe. There's lots of different things potentially in here as well. I'll come to you in a second. Yep?
I'm not really sure, but aren't like the Neanderthals like the first type of humans?
So, again, you should come to my university. We discuss all this sort of stuff in my lectures. Weren't the Neanderthals the first type of humans? What's a human? How far back do you go? We were talking the other day, weren't we, about this, and some of my other students are here, about the very first things that were in our lineage. So about 7 million years ago, Sahelanthropus was walking around, this thing that looked a little bit like a chimp, a little bit like a person. Is that still an ape? Are we just apes? Then we get to things like Lucy, the Australopithecines, around about 3 million years ago. She was walking around, she was making tools, she was doing all these weird and wonderful things. She's not a human being, in the same way that you and I are, but she's not just an ape in the same way that a chimp. Either we accept that we're all just apes and just animals, but if we think we're special, where does that special stop? So I would say, going well back to things like Lucy, she was a person. She wasn't a human, but she was a person. She was part of that journey. By the time you got to things like Neanderthals, they were definitely human. They weren't human being as a species in the same way that we are, but the moment you get to the Homo group, so Homo erectus, Homo habilis, about 3 million years ago, 2.5 to 3 million years ago, they were people. They were people, they were humans, they were that human journey, but there were multiple species of humans. In the same way we've got multiple species of big cats, multiple species of chimp-like animals that were chimps and bonobos, we had multiple humans living at the same time. So we had Homo erectus, Homo neanderthalensis with us, with Homo denisovans, with floresiensis, these little people in Indonesia. They were all human beings, but not Homo sapiens sapiens, the same way that you and I and the rest of us hopefully are. Yes?
You said we're not just Homo sapiens, and that's cool. Is it possible to not be just Homo as in the genus?
Yeah, so at what point do we just describe ourselves at a different level? Do we just say, it doesn't matter if we're Homo sapiens, we're just the Homo group, so it doesn't matter if you're Homo sapiens, Homo neanderthalensis, whichever. It doesn't really matter. It doesn't really matter if I say, would you mind if I said, actually, we need to reclassify you as a chimpanzee?
I'd be...
Would it actually upset you? Would you be...
No, I think I'd be quite pleased.
Yeah? Actually, in my mind, I think we should be, and other people, Jared Diamond, for example, have said before that we should be another species of chimpanzee. We should be the third species. We've got Pan-troglodytes, the chimps, Pan-paniscus, the bonobos. There should be, potentially, Pan-sapiens, the other chimp, which is just us. So it depends whether you look at our genetics, it depends whether you look at our culture, whether you look at our anatomy. It all depends on how you look at these things. And ultimately, someone has to decide that. So science is never neutral. There's always politics and feelings in science. Yeah?
Can we talk about the ocelot?
We're going to talk about the ocelot, sorry. Go.
So we're starting to peel back the skin layer now. So you can see some lovely red tissue here. What tissue do we think that is? Hands up if you know what that is. The lung. No, it's before we get to the lungs. So all we've done is just taken off the top layer of skin. So what do you think we've got to here? Second layer of skin? No, so actually the whole, even though there are multiple layers of skin, that's all contained within this small piece here. So actually it's quite thin, really. Fat, no, but we can see fat. You can see lots of fat here. This is fat. That's fat. Really super squishy. You can press your finger into it and it makes an indentation. It's sort of like plasticine or what's that? It's a bit like slime. What? Play-Doh, that's the word I was looking for. Doesn't smell like Play-Doh though. And actually this animal has got quite a lot of fat, which makes me think, you know, it wasn't starving when it died. It was still eating and doing quite well. Fat's it's a really weird material. It's got a really strange feeling and it leaves sort of an oily sheen on your gloves. Sometimes we have animals come into the vets that have absolutely loads of fat and it sort of covers the whole thing and it makes it really, really difficult to do operations and things like that. This one doesn't have loads in a sort of worrying way, more in sort of a reassuring way that it was doing quite well. It had stores of fat for if times were hard. So we've got the skin. We've got something called fascia, which is connective tissue, which you can see there. It's sort of that, almost looks spider webby. Can you see that there? Where are we in? Can you see it? I don't know what you can see. So it's this stuff here. Can you see? It's really translucent, but it's what holds everything together. Keeps it all sort of sitting where it should. Can you see how that's sort of thin and... Anyway, I love fascia. Breaking through it is really quite therapeutic. Anyway, back to this red stuff. Anyone got any idea what it might be? Liver. We're not into the abdominal cavity yet, so it's not any of the organs in there. It's not liver, it's not kidneys, it's not lungs. Muscle, yeah, that's muscle. So these are, and it's actually got quite big muscle bellies under here. So we're going to sort of reflect the skin a little bit so you can see those muscles a bit better. Catherine's doing an absolutely beautiful job of that there. Yeah. Got a beautiful muscle there that I might try and expose as well. I don't know where he's gone. Has anyone got any questions? Yeah? Does the fur keep the animals warm? Yeah, definitely. That's one of its, apart from camouflage, that's sort of its other main function. Most mammals are covered in fur, and it's definitely got an insulating function, but fat has a really insulating function as well, which wouldn't, you know, sometimes that's a negative. It can keep animals too hot if they have too much of it. But yeah, fur and fat work together to keep the animals nice and warm. Sorry? So the question was, will we take the internal organs out? So sometimes it can be easier to remove them from the body and to lay them out so we can see them more clearly. I think we probably will definitely remove the sort of the liver and the digestive tract so we can. So last year, were any of you here at the llama dissection last year? So last year we got the intestines out and they were super long, like crazy long. And we stretched them out. And if you think about a llama, most of its body is actually a big, big belly, housing all of that intestine and that digestive tract. Whereas cats, they're pretty thin here, aren't they? Not a lot of space. So do you think we're expecting to see a big, long digestive tract or a shorter one? Probably a shorter one. Well, we'll find out soon. It is a smaller animal as well, but even if the llama was this size, say a llama was this size, still that much of it is going to be belly. A bit like a cow, they have those big, huge bellies to house their stomachs and their intestines because they have them. And why do you think that is? Why does a llama need to have so much more digestive tract? Because it's herbivorous, exactly. And why would that mean that it needs to have a longer digestive tract? Perfect. Firstly, what does herbivorous mean? Someone else? Yeah? You're a herbivore, so you eat plant matter. And exactly, as you said, plant matter takes a lot more energy, takes a lot more digesting to get the energy out of it. Whereas, what do you think these guys are eating? They eat meat, they eat other animals. So this one's probably eating things like rats, mice, baby capybaras. So they eat flesh, they eat dead animals. And it's much, much easier to extract energy from meat than it is from plants. So they don't need as much digestive tract, but we'll get to that. We'll just get this skin off first, and then we'll get inside. I don't know. Let's have a look. Yeah, maybe. Let's clip a bit of the... So Catherine's just asked me, there's some sort of dark patterning on the inside of the skin. Catherine's just asked me if that's the pattern of the fur. And cat's skin is generally patterned the same colour as the fur on top, so it probably is showing through, but I was going to see if we could clip the patch. So the cheetah, when we shaved the cheetah, it had perfect spots all over its skin. It was amazing. It was really cool. But I don't know. I don't think that does look. I don't think it is. I'm just using the scissors for this bit. This is called blunt dissection. It's where you can either use your finger or something that doesn't have a sharp blade. And because the fascia is really useful to hold everything together, but it's not particularly strong, you can push your finger through it or a blunt instrument. And it's a really good way to get through the layers without a blade and making sure you don't damage anything inside. So I think that's pretty good. Yeah, because that's a muscle layer there, so. Actually, we could, if we dissect out the scapula, not the scapula, the clavicle, then we can see the vestigial bone. Ben might want to talk about that. Where's he gone? Yeah? I don't know. What are we going to do with the fur? I don't know if we have any research projects about the fur, so I'm not sure. I don't think we have a specific plan yet, is the answer to that question. So normally, to be honest, normally if I was doing a dissection of an animal that had died of something that of a known cause, so like I knew it had been put to sleep, I probably wouldn't do this bit. I would just go straight into the abdominal cavity and look at the organs. But because this is a learning experience today, we're exposing the muscles and things like that so that you can see the whole body. But normally, probably I'd just go straight into the abdomen, I think. So Ben, I was thinking if we dissect out the clavicle.
Yeah. So as I mentioned earlier, this is taking longer than a lot of people expect. So I can imagine at least one or two of you going, oh, hurry up, I could cut it quicker than that. This is why science is so fun. We spend so much time very gently and very delicately going through. What Jess and Catherine are doing is a little bit like being science detectives. So I love reading my detective novels and spy novels, and in my spare time would love to be a detective and solving crimes. But actually, we're kind of doing that thing anyway, because what Jess is doing is looking for any tiny changes, any tiny differences within the anatomy. Is the color of the muscle different on one side? Is there a sign of infection? Is there one broken bone that might explain something? Which is why we're taking slightly longer than a lot of people expect when they turn up to events like this. And what Jess is doing now, she's peeling back this lovely layer of skin here. You can see the fat underneath that she mentioned before. And actually, it's coming away quite nicely, isn't it, Jess?
Yeah, it's really beautiful.
Yeah?
Coming really
So
well.
Jess is now exposing the...
I'm just going to expose this limb. So we've got sort of one side of the body without skin, so you can see what it looks like at the muscle layer. And then we're going to go inside to the bones here, which I imagine you'll want to speak about, will you? Yeah.
Do you want the other side done?
I don't think so, because
No?
it's the same. I don't.
Do you want this taken back here, or...?
It's up to you. If you want to get involved, put some gloves on.
Yes.
I'm not going to come up there, don't worry.
If you have any questions about the skulls at the front, we've got Emily and Ellen here as well. They're going to put their hands up and wave at you. They're at the front. They are also able to talk about the different skulls that we see here as well. And some of the nice things that we can talk about are the different shapes of teeth, the different size teeth as well. So this is a cat that's alive today. Well, this one isn't. This is a model. But this is from a type of cat that's alive today. We've got an animal that looks very similar to a cat, with nice sharp teeth at the front and big carnassials at the side, but is not a cat. And we've got some things that look like it's from a vampire film, but actually this is walking around Norfolk right now. Any idea what this thing is? Anyone? Yes. Not a muntjac, nearly. Yeah. You can't remember its name, but it's got sharp fangs and it is a deer. A water deer, yes, this is a Chinese water deer. Looks really cute and cuddly when you see them on the films and photos of Norfolk Wildlife Trust, for example. These things have massive canines. So I wanted to start thinking, why do these plant-eating animals have canines as well? And how does this all link to the cats that we're talking about today? Do they eat cats? They do not eat cats, no. Why do you think you might need a really big set of canines if you eat just vegetables and plants and things like that? Why do you reckon? Because they're tough. We're not going to take this skull out now, but as Jess said earlier, we're going to remove the bones at a later date, because this takes a long time to do. So we will clean the bones up. Catherine and I have a very special lab called the Death Lab. It's very cool, where we've got tanks of insects that strip meat off bones. So if you left your hand in there for a weekend and just sat there like this and came back on Monday morning and you're like, oh, what time is it? You'd have a skeleton hand with a watch on. They don't like live flesh. Well, I'm quite old, so mine's barely half the time. So we've got a death lab where we look at these animals and strip them down. So we'll be doing more of that at a later date. Yes? Yes, so with the Chinese water deer's teeth, for example, they don't have antlers, so they're showing off in a different way. It's territorial behavior, and they fight and use their tusks of their teeth to fight one another. Very cool. So we're just, as Jess has said, Catherine and Jess are taking the skin back on that side. They're taking one of the limbs out.
Let's get this limb out and then...
Why isn't there blood everywhere? A lot of people come to these events and go, oh, there's no blood. I want blood everywhere. Where is the blood? Why is it not spurting out and Jess covered right now? Yep. Oh, we're still on the outside. Yes, that's one good answer. Once I cut them...
if we cut its skin like this while it was alive, do you think it would bleed?
It would bleed, but you wouldn't be covered just yet, would you? I mean, maybe it's because we're not fully... Maybe when we open that, it's just going to wash this lot in red.
Yeah, maybe.
Maybe, we'll see.
Maybe they want to take a
Keep
step
your mouths
back.
closed in about 3 minutes, you lot. Why else? Why are we not covered in blood right now? Yeah. The heart's not pumping. Yes, the moment the heart stops pumping, it's not going to be squeezed out and pumped around. The blood starts to decompose and solidify. So a lot of people say, why are you not covered in blood when the animal's sadly dead? Nothing's pumping that blood around.
Shall I dissect this out and then you can talk about that while I'm
Yep. What are you taking out?
clavicle? So you can talk about how it means it can reach.
Yep.
So you can see we've got down to the muscle layer. I'm just going to find a very special bone inside here for Ben to tell you about while we're... Where are you?
So that you know where it is, where is your clavicle? Or where are your clavicles? Anyone who's not answered the question yet? I've got some adults doing this and pointing to various bits of their bodies, which is hilarious. Yep, pointing to your throat, pointing anywhere else, pointing slightly off from your throat. And yeah, near that shoulder bit. Your clavicles are your collarbones. We have these lovely big collarbones that sit almost like coat hanger pieces from there to there, and it anchors your top of your arm together. Having a collarbone there keeps your whole shoulder nice and stable. So when we used to swing through trees as our ancestors and great apes, having these strong clavicles allowed us to swing and swing and swing and kept all that as anchorage. Now, when you have a big set of paws like this and you're chasing someone and you just throw your paw around to try and catch them with these huge claws at the end, instead of this nice stable joint, you want something really loose. So I can't do that. I can just get my arm over in a nice arc, but I can't suddenly make it go really dynamic and move in different positions like this big cat can here. And you notice this shoulder moves in so many different directions because it's not attached in the same way that ours is. It's much looser. So Jess is exposing the clavicle here. And in ours, it's massive. It's a huge anchor point. And our bones aren't just there to keep our muscles attached. They're not just there for stability and keeping your skeleton together. Sometimes these bones are anchor points where muscles attach, and they're really useful for allowing different types of movements. If you've got really big collarbones, really big clavicles, it means there's lots of anchorage. So do you reckon the collarbones, the clavicles in our cats are big, or do you reckon they're small? Yeah, big or small? Big? Small? What do you reckon?
Middle?
Big? Yeah? You reckon they're really small? We've got some people saying really big, some people saying really small. Remember, my clavicles, my collarbones go from the middle to here. They're massive for an animal of our size. Lots of anchorage.
What about showing with their hands how big they think?
Yeah, how big do you think? So mine are about this size, from there to there. How big do you think the collarbones, the clavicles in this ocelot are going to be? We've got someone saying that size, that's pretty small. Someone saying that size, someone doing that. You're all about here. There's quite an agreement over here. We've got one person saying about that, and she's now concerned she might be wrong. Well, this is the smart area, but there's no point in you suddenly doing that now I've said that. Yeah? They've all just gone. They are tiny. So Jess has just exposed the clavicle here. I'm not sure if we can even get this, it's so small. It is against my glove. It is an absolutely tiny clavicle. So look how happy you are that you were right. Smarter than the kids, finally. We've got them. Losers. I don't mean that. It's a tiny clavicle, a tiny collarbone. And being so small, it still offers some level of anchorage and support here, but it's not stopping that amazing mobility, that amazing movement in those shoulder joints. So when you're chasing with a piece of feather or a piece of string with your cat, and suddenly its arm is doing all these amazing, wonderful things that you can barely watch. Part of that is because the clavicle, the collarbone, is so small, it's allowing it to move in ways that we couldn't possibly even begin to try and do. Yeah?
If you hold that slightly.
In us or them?
Oh, I'll just cut that bit off.
So does a clavicle usually snap? Jess, what happens if a clavicle snaps?
The thing is, it's buried deep inside this muscle, so it's really, really protected. And actually, the only clinical significance of a clavicle is that sometimes you see it on an X-ray and you're like, what's that little line there? What's that? Because it looks like it shouldn't be there. But no, nothing ever really happens to it. You wouldn't break it. It's literally buried in this huge, I mean, you can kind of see it here. So I've cut inside this muscle belly, but it actually sits inside there like that. So it's in a big cushion. You'd never break it, and the arm's held like that, the leg is held like that.
I think it's really important. So when you've got a bone in a museum, it's this long, hard thing that's white, and if you tap on it, it echoes, and if you drop a bone from a museum, what happens to that bone if you drop it on the floor? Smash, you get in trouble, you get thrown out of the museum, yeah, all these things. What happens if you drop one of these bones from this animal? If we took the bone out now from its femur, its thigh bone, and dropped it onto the floor, what would happen to that bone? Would it still smash?
You have a go. You're better.
It would literally just roll around. Yep. So bones from animals that are alive or recently dead are full of different other pieces of soft tissue, of blood, of different vessels. And because bones are living things, you don't see them snapping in the same way that a skeleton's bone would from a museum, for example. So hopefully they shouldn't snap. If we snap anyone snapped their clavicle in here or broken their collarbone? If you break your collarbone, it's meant to be very painful. And because ours is so big and so important for anchorage, it can take a while to heal. You often have need an operation to sort them out. Whereas Jess has said if they break theirs, it's not such a problem because they're not using them in the same way that we use ours. Yeah. Because of what? Yeah, so the reason, so evolution chops and changes. Evolution doesn't think about doing stuff. Evolution's just, it changes if something benefits the animal or the species or the group, whatever. And by having, at some point in their past, we looked in the fossil record, we could probably find that point when the clavicles, the collarbones, started to become smaller. And then after that, once they became smaller, you'd probably notice the claws became longer and sharper. And you see this snowball effect where if it's working and they can be slightly more mobile, so I'm sneaking up to Catherine at night, trying to stalk her with my coloured coat and all these wonderful things, and I'm blending in, and I've got my soft feet that she can't hear me, and I suddenly come and chase, and all I can do is that from the side or up or down. I got a chance of catching her, but if I've got slightly smaller clavicles, then maybe my arms can bend in ways that I couldn't before as a species, and I've got an increased chance of catching her. And then I'd pass that on to my kids and their kids and their kids. So by having smaller clavicles allowed them to hunt more efficiently, to move better in the trees, and to do different things. Yeah. Oh, that's a good question. Jess, it looks like there's less fat on the Jaguar than it does on the Ocelot. Is there a reason for that, or is it just...
So we did take some fat out of that one, but actually I think that was a less fatty individual. But I mean, at that position on this one, there's not really any fat either. This one's fat was all around there, and that bit's gone of that one. So, but it's not really a species thing generally. I mean, certainly some animals are more fatty, some species are more fatty than others, but it's also a very individual thing. So some animals carry more fat than others. Some of the cats that I see at the vets are really, really skinny and really slim, and some of them come in and they weigh 10 kilos and their bellies are like this. So it's a very individual thing as well, and these two individual animals were just different in the amount of fat that they had, that they carried on their bodies. Yeah. No, it was put to sleep. It was euthanized because it was getting really, really sore. It had really, really bad arthritis. It couldn't walk anymore. So, yeah.
Right.
Okay, so I think that's about as much of the skin as we're going to take off because we've kind of got the impression. So you can see that under the skin, we had some of this, we had some fat, some of this fat. We had some fascia, some connective tissue, and then we had all these layers of muscle. Can you see that as well? And they're quite thin layers. Underneath here, yeah, they're the ribs. All working in slightly different, in different ways. Their fibres aligned in slightly different directions. And here, you can see where it needs the power in its limbs. So the power from those comes from these pectoral muscles here, and also the muscles for biting as well. All of that musculature in the neck needs to be really, really thick. And around these arms here as well. And then you can see in the paw here, so hands and paws are super complicated because obviously you need to do lots of movements with them. So you've got lots of muscles, lots of tendons and lots of ligaments in your hands. And sometimes, if I can work out which, you can sometimes work out which, I need to get that off really. Hang on. You can sometimes make them move their claws by pulling the, let me just see if I can do that. And this one. I just want to get that claw out. Could you just slice on top of that? On top of the claw? Yeah. So fiddly. Once you've gone through that connective tissue, it should just pop out. That's it.
Yeah.
But all the way along. Oh, that's not going to work. I think we might come back to that at the end, and we'll just move on to the abdomen. Also, oh yeah, you can start to see how the inside of this cat's head, without the skin, starts to look like those skulls over there. Sort of as you peel away each layer, you get sort of more of the idea of the skeleton beneath. Okay, we've done clavicle. I think we might move into the abdomen. What's the time? We'll come back to that, I think. Where's that penis gone? Yeah, I think so, please. So I'm going to move into the abdomen now. I'm going to start to see some organs. So I'm going to go through a line here, which you can just about see. There's a white line in the middle. Can you see that? That's called the linea alba. If I was going in to operate on a cat, that's where I'd go through, because that's where the muscles meet. There's no nerves there. And so it's the least painful place for me to cut. Yeah? So if I was doing like an operation, you mean? So if I was doing an operation on a cat, first of all, it would be prepared totally differently. It would be shaved. Everyone would be in a sterile operating theatre. We'd all have washed our hands really, really thoroughly, so there was no dirt on us. We'd probably have headgear on, masks on to prevent infection. It would obviously be alive, so it would be breathing. It would have been anaesthetised, which means we would have given it some medicine to make it sleep. It would have a tube down its throat, so we would be doing the breathing, or helping it doing its breathing. We would put a tube down its throat to give it oxygen and to give it gas that continues to keep it asleep. It would have loads of monitors on it. Someone would be monitoring its heart, its blood pressure. Someone would be checking the carbon dioxide that it breathes out to make sure that it's doing well. I wouldn't obviously do this. I wouldn't take all the skin off of an animal that I was operating on. I would just, depending what I was doing, I'd probably make an incision up to that big. I mean, obviously every operation's different. You'd do it as quickly as possible, and yeah, the conditions would just be completely different. But you'd still go through this linea alba because you'd want as little bleeding as possible and as quick a recovery as possible. Which one's the name? They're both new. How long could you leave it open for? I mean, ideally, you don't want to be operating for more than a couple of hours. The infection risk increases, but also when animals are anaesthetised, it's much harder for them to regulate their body temperature, so they can get really, really cold. Although we do things like we have heated tables, and we do lots of things to try and keep them warm. Ideally, you don't want it to be more than a few hours, really. Oh, that's true, actually. So a lot of people do flank incisions for cats. I actually, yeah, some places do it midline and some do it flank. It's easier to access the ovaries, which are up here, which is the bit that you're taking out. Some people think that it has a better recovery because the organs aren't onto it, but to be honest, it's an area of disagreement between vets. And to be honest, some people are trained to do it that way and trained to do it that way. And ultimately, as a vet, the safest procedure you can do is one that you've been trained to do and the one that you've done the most times. So, yeah. And in a cat, because they're so small and their body weight is so small, it makes less of a difference than in a dog. But in a dog, we always go through there. In this country anyway, but in different countries in the world, they do things differently. So that's just what we choose to do here. Sorry. Okay. Oh, it's very fat inside as well. Wasn't expecting that. Right, so I am through the abdominal wall now. You can see I can lift that up. And there's sort of a cavity inside here. That's where all the organs are. So this is a layer of muscle. And it's got a lining on the inside as well that prevents any of it coming out. And inside there are all of our organs, which are all covered in loads of fat. Oh my goodness. Wow, look at that. Oh, and there's the mesentery. How far up shall we go? So let's go up here. Okay. Oh, you might, yeah, can you smell it?
So we just got a quick question here. So the lady here asked, what was the bit that you just showed us, Jess? The mesentery.
So this is the connective tissue that holds all of the intestines together. And it's also where the blood flows through and also the lymph. So that's all, it's really beautiful, isn't it? So that's the intestines. And much, much, much less. Do you remember when we opened the llama, the people who were here last year? The first thing we saw was a big balloon, wasn't it? A big, huge stomach. And actually, can't even see the stomach of this one yet. Look at all that fat.
So the question there, Jess, is an interesting one. So the fact that there's fat in there, is that a good thing, showing it was well fed? Is that a bad thing, saying it was overly fed? Do we go, well, it can't have been starved because it's got fat there? What do we...
I don't want to offend anyone here, but in the animal kingdom, old males do tend to get quite fat.
Don't keep looking at me when you say old males get fat. That's a little bit rude there.
So, I mean, you know, for a 15-year-old cat, although, you know, cats have a tendency to go one way or the other. They either get really fat or they get really skinny when they get old. So.
Has anyone got a fat cat at home? A few of you get straight away like, yep. As Jess is saying, the old boy cats typically get a little bit fatter than the...
Although, to be honest, that's usually if they're castrated, but...
Okay.
How thick?
How thick? How thick? Well, the fat layers can build up, as Jess is saying.
I mean, look how thick that is.
That's quite a lot of fat for a little cat, actually. And we're going to start putting some of these things in trays that you can have a look at a little bit more closely. That we're going to be passing different things around. That's what fat looks like. Fat is the same substance that we get if we're having a big Christmas dinner and we don't do much exercise for a few days. Or weeks, some of us. But it's the same thing. And fat's really important. Fat is really good for us, isn't it? It stores a lot of the energy we need. There should always be. I mean, this is an amazing predator that's evolved for millions of years to kill and hunt and be efficient, and it's still got fat in its body. So some fat is really good for us, and we need it. Too much fat puts too much pressure on our hearts, on our lungs, and different body parts. So it's finding that balance within nature. A few more hands up. Yep.
When you took the first layer of fat out, and then took that bit,
Ah, that's, you lot are really, I've got to say, you lot are really good with your observations and the questions you're asking. So the question there, Jess, is there are different types of fat coming out. The fat we're seeing around here is different to the big, thick, lumpy fat we're seeing on the inside there. Is there any differentiation between that? Is there any benefit to that? Do we see different levels? So my answer, without knowing fully the answer to here, the fat that's sitting under your skin is moving much more. It's much more dynamic. It's being spread out, so you're seeing this more sort of a surface fat. The stuff you've got in there is just big fatty deposits that's sitting in the cavity here that's filling space, that's not moving as much. It isn't twisting and turning as much as the fat will on the skin in somewhere like that. Yep, what's your question? What's the greenish thing? These are parts of the questions I love in this thing. Yep. Why is this, compared to humans, why is the fat slightly pinker rather than yellowish? So when we look at our fats, so primates store different types of fats in different ways, and the chemical compounds are slightly different. So the fat that we see in big cats is metabolized much quicker, it can be burned off much more quickly. Ours just sits there as sort of, once that fat is there, it's there. It's big stores. Primates store fats differently. So I've dissected a lot of primates and people, and our fat looks very different in some parts of our body. So the fat around here will look very yellow, will look quite different. The fat in our thighs, for example, is a different type of fat. So it depends where the fat on the body is. It also depends what species or what group of species you're looking at as well. Any more questions? I know you two. Yes, go on, you three. You lot are amazing with questions today. You're going to have to move your thing because I can't hear you mumble, I'm afraid.
You said that you got it.
Yep.
How was that?
How was that? It was better for me than it was the human. So yeah, in my mind, as a biologist, I work with different animals, and humans, we were all just another type of animal. Jess, what are you up to?
So I wanted to show you so this part is called the thorax. Things inside there are the lungs and the heart, and it's at negative pressure. And I wanted to show you that when I cut through it, there was a puff of air that came out, but I accidentally cut through it. So anyway, we have got a layer of muscle here that separates the abdomen from the thorax. Does anybody know what this is called? The diaphragm, exactly. Anyone know what the function of the diaphragm is? Hiccups.
Do you have a diaphragm? Yeah? Okay, good. Yeah, you've all got a diaphragm.
You go with that and I'll get the digestive tract
So
out.
your diaphragm is...
Actually,
okay.
just before I do, I'm about to take the organs out that are inside. So while Ben talks to you about the diaphragm, let me just show you the diaphragm there. So you can see it's right up inside. These are the ribs here. The diaphragm is curved up here. And actually, the chest cavity is only that big. So all of this is abdominal organs. I'm now going to take them out so that we can look at them more closely. But just so you can see, these, when I first opened it, they sort of all sit like that. And that's what the inside of the cat looks like. Okay.
So first of all, your diaphragm, as Jess is showing you really nicely there, it stops all your abdominal organs slopping around and going into your chest cavity. You don't want that. If this cat is stalking down a tree and trying to stalk its prey, and suddenly its stomach falls up near its chest, then it's going to fall where its lungs are, where its heart is, and it's going to cause lots of damage, and it might even cause injury for this animal. So first of all, it's a physical barrier to stop the different parts of the internal organs slopping around near each other. But importantly, when you all take a nice deep breath in, after 3, 1, 2, 3, go on, and release. How are you breathing? What's doing that? Is it the ribs breathing in? Are they just deciding to expand? Is your heart doing something? It's all your diaphragm. Your diaphragm is moving and pulling the air in, and it's releasing and pushing it out. It's the set of bellows that's helping your chest do its thing. Your diaphragm is what's pushing and expanding that chest, contracting it. So it doesn't look like much. It looks like a little layer of tissue that's not really doing much. Without your diaphragm, and without it functioning correctly, we wouldn't be able to get those nice big breaths in. So again, one of these parts of the internal anatomy that's doing lots of different jobs at once. So Jess is now cutting through that. So she's separating, she's breaking that barrier between the abdominal organs and the thoracic organs. Who knows what sort of organs you might expect to see in the thoracic cavity? What am I expecting from there up? Yep. Hearts and lungs, yes. So you should see We hope. There's one heart and a pair of lungs in there as well. So that's what's up in the chest cavity. And then down in the abdominal cavity, you've got things like your stomach, your liver, your spleen, your mesentery, your small, your large intestine. All the digestive things are further down. So you've got some cable ties now. Jess, what are they for?
Well, the stinkiest bit of the dissection is opening the digestive tract. And so I try to minimise that, and also to minimise spilling digestive contents everywhere. So normally when I do a dissection, I would cut this open and have the whole thing open so you'd see the whole body. But because we're preserving the ribs for the skeleton, I'm sort of going at it a bit backwards and cutting sort of up through the bottom. So you've got 2 tubes running down from here, which we will see in a bit. I'll dissect them out. Do you know what the 2 tubes are? Perfect. One's for food and one's for air. Does anyone know the names for those? Trachea for air and esophagus for food. Perfect. So the esophagus, the trachea and the esophagus run here, through the neck, through the neck. The trachea goes here to the lungs, and the esophagus keeps going through the diaphragm. And where do you think it takes your food to first? Your belly. Which organ in your belly do you think? Stomach. Perfect. So, because I can't pull the esophagus all the way through and tie it off, I'm just going to tie off the esophagus here so that we can take out the digestive tract without any of the stuff that's inside it falling out. So that's what the cable tie is for.
How do you throw up?
Sorry?
How do you throw up?
How do you throw up?
Yeah.
So it uses your abdominal muscles and the muscles of your stomach. They contract and push the contents back up through the tube. So the action comes from your stomach and from your... And you squeeze it. Exactly like. Perfect analogy. Well done. That's awesome. Yeah, that's exactly right. Which is different to regurgitation. Well, this has got a bit vetty, but if you eat some food and then it hasn't yet sort of got into your stomach and been digested and forced up like that. Regurgitation is sometimes when cats, they eat a bit and then it comes straight out and it still looks like food. I don't know if the cats have done that. Anyway, vetty thing. Vets love to ask you questions about exactly how your animal was sick to check if it was being sick or if it was regurgitating. Having such a glamorous job.
It is great. It is great. A couple more questions. Yes, get your hand up. What's your question?
Do you have any ideas of what it could be?
Of what could be? Do we? So with this far through, I guess we're about two-thirds the way through the necropsy now.
Yeah.
Anything obvious in terms of what was wrong? We know that the cat was euthanized, so it was put to sleep.
Yeah.
Any idea why, in terms of any disease in there, any infection that we're seeing so far?
No, it all looks great so far. All looks normal.
And I guess sometimes we might get to the end of this and go, we still can't tell, but it might be microscopic. It might have been something that was just not quite right somewhere. It might just be it was a very old cat. And the nice thing is, even if Jess comes at the end of this and says, we can't see anything that was actually wrong with this cat, but
That's quite good.
Yeah, we still know it was arthritic. It had changes between its bones. It was making it a little bit more tired
If it was
and
too
achy.
sore.
Yeah, it might have just
animals...
been it was just an old cat that was its time to go. But at least we know that. At least we'll be able to tell, or Jess can tell the zoological collection what wasn't found. What we don't find is almost as important as what we do find.
Yeah, definitely.
If we got in here and we went, oh my goodness, there's like a terrible infection that was burning this poor cat up and it was really suffering, then I think questions would be, well, why didn't you notice that? But actually, it almost might appear there's nothing more this zoo could have done to make sure this cat had a very happy life whilst in captivity. Little girl had a hand up at the top. Yes, can you be nice and loud for me? How did it freeze? I love that you did the freeze. How did it freeze? We stuck it in a freezer. So when an animal dies, these sorts of animals, after a little while, they become very stiff. So if you put it in a freeze position, it would be in that position for about 24 hours, so one day. And then as the acids and the different chemicals in its body start to break down, it becomes all floppy again. So it's something called rigor mortis. So at some point, we couldn't have moved this lovely Jaguar's leg around like this then after a day or two, we can start to move it around again. We put both of these big cats in big cat, small cat, in a freezer, but at the moment it's laying in that position because when you lay on the floor, you just flop out, don't you? Like flop out, nice and flat, like a happy little human lying there. Cats don't lie like that because their bodies are slightly different positions and their chests are different and their legs are in different positions. They lay in a slightly different way. So a cat can never put its arms out straight and its legs out straight like a star. A cat can never do that. Never try and make your cat or your dog do that because their bodies are slightly different to ours. Okay, Jess, what have we got there? What's going on?
So this is the digestive tract minus the esophagus, the tube that comes from the mouth. So actually the digestive system starts here. We talked about the teeth and the tongue. You know, that's where the food processing starts. And then the esophagus is basically just a tube. It's transport to transport it down to the next bit, which is the stomach. We've got the intestines. We'll talk through those in a minute. And then this bit here is kind of the other end, the esophagus. So this goes all the way through here, and then out to the anus here. So this is the rectum and the colon. So obviously, to have this digestive tract and to be able to lay it out and show you, I'm going to have to cut this end as well, which feels empty, so I actually don't think I'm going to cable tie it.
Oh, you've had your hand up a long time. What's your question?
Underneath the pile of organs, there's a red thing. I want to know what that is.
There's a red thing.
Under
the pile of organs, there's a red thing.
We'll get to it. We'll get to it. So,
what do you reckon it might be? Oh, either the stomach or the heart. Good guesses. I'm not going to tell you what it is yet. Or nor is Jess. Oh, don't tell us yet.
basically, at the most simple level, the digestive tract is one long tube. Comes in the mouth, goes down the esophagus, through these various parts of the tube, and then it comes out through a tube here. So this is the colon and the rectum. I'm just checking nothing stuck in there that would prevent it. But no, just a bit of poo.
Just a bit of poo.
Just a bit of poo. I'm going to get some clean gloves.
And that's as simple as that. You are just one big walking tube. It's a different way. You might think you're special, that you can do all these wonderful things. You can play an instrument and you can play games on the computer and do all these cool things. You're just a walking tube with a mouth one end and a bum the other. If you understand that, you are great at biology. And all of these animals are exactly the same. Whether there's a whale in the oceans or a bird in the sky or your ocelot here or even you, it's a mouth one end, a bum the other. And it's just different bits in the middle, whether it's got legs or fins or wings or whatever. But that's the important thing. Energy in, energy's used to allow it to do its thing, and then coming at the other end. Jess, I noticed there's some stuff going on down here, and your face shows that you...
Well, as I put my finger in the anus, I felt two large lumps either side, which is normal. They have glands there.
Yeah.
But they're very, 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 very full. And I've tried to squeeze the content out, and it's so super hot. Oh!
Do you want a mask?
Look, look, look.
So for those of you thinking this smells quite bad already, I'm going to warn you, you ain't smelt nothing yet. This is why Jess is one of my absolute best friends in the world.
I used to wash her hands before she can't touch them.
I've got no sense of smell, so I'm good with this. So Jess has noticed that the scent glands around the bum are quite full. If you've got a cat or a dog at home, they have specialised glands, little sacs at different parts of their body. And some of the main sacs are just either side of the bum, and they're meant to be there to smell. It doesn't smell in the... It doesn't smell in the same way that a fart does. It's much worse, isn't it, Jess?
Wow.
Yes.
The other bit that I smelled wasn't too bad. I thought, oh, wow.
Do you want to
Catherine?
Do you want to... Anyone want to smell? Can they smell? Are they
I
later?
don't know.
Yeah, it's all right. Parents are here. It's up to them to allow them to do it or not.
Don't get too close, but just sort of...
So some of this can or was historically used in perfumes to fix the scent as well. The reason these animals smell like this isn't just because they want to be a little bit stinky. It's because they are marking territory. They're showing what's going on. So not every animal in the world uses Facebook or Twitter. That's just us. But other animals want to tell each other what they're doing, how they're going, how they're healthy or not healthy. And a way of doing that is through scent, through smell. And having scent glands like this is a really good way that the cats are able to tell other cats in an area who's around, how healthy they are, how their abilities to reproduce might be. So whilst this smells bad to you, to another cat, this is like a social media post. Jess.
Sorry, I wasn't really listening to it. I was a bit focused there. It was this really musky smell.
Yeah.
I don't know if you just said this, but it's sort of an oily substance which they leave for scent marking. It stays for a really, really long time. And I mean, you can see it, once it gets stuck on something, it's not going to come off.
So a lot of these cats will mark on trees or bushes, and as Jess said, it's oily. It sits there. If it's just watery, like pee, when you have a pee, it just washes off really quickly, and if it rains, it goes. This stuff is like a glue that sits or stays there, because if you're walking, some of these animals might have a territory that's 100 kilometers, and you don't want to do that scent marking 5 times a day around every part of that territory. You want something that's going to be there. Again, like a social media post that's going to be there for a week or even a month maybe in the right conditions. So this is, I keep saying this whole idea of a social media post that tells everyone who you are, where you are, how you're doing, whether you're big and tough or maybe not so big and tough. By having this oily scent, this liquid, this gel-like substance allows these animals to tell all their mates and their enemies just what's going on. So we've looked at so is there anything, I mean, they were full. Was that a problem? Was that a cause of death?
I don't really know, because when I cut it open, I don't want to touch it again because I just put clean gloves on, it didn't look like it was full of purulent, pussy material. It looked like the gland itself was just really big. And I don't know what's normal for an ocelot, because I've never felt an ocelot scent gland before, so.
But now you have. And you do see, and we do see this in cats and dogs in your vets, for example. Sometimes some individuals will come in with slightly larger glands, slightly more full glands, but it doesn't usually cause death or a problem.
No, so it can give them a bit of an itchy bum, and it can make them do the bum rubbing thing.
Yep.
But the anal glands in dogs are slightly different. They're more sort of lubrication than for scenting purposes. Don't know. I'll write it in the report. It definitely wasn't what it died of, but...
But like Jess said earlier, this is a really nice example. Jess hasn't done this before because very few of us in the world have dissected an ocelot. So yes, you're here at Norwich Science Festival and we're seeing another dissection, and you guys kind of get used to this idea that we just do a dissection every year. But actually the things we dissect, sometimes maybe this has only been done 4 or 5 times in the whole of Britain. So even by Jess saying, I don't know whether that was normal or not, isn't because Jess is rubbish. It's because no one knows. But actually, even having this now will allow us to have this idea of what's normal, what's not normal, what can cause injury or death, or what might be an acceptable level of slightly full scent glands.
And what I can do is I can measure it, so we have, an actual measurement, however many centimetres. I can write that in the literature, share it with other zoo vets, and then they will know if they find an ocelot, well, that was within the range of these other ones that we saw. So, ooh. It's got something in its stomach. Right, so we're going to lay out. Look at that.
Before you do, can I just have a quick chat about this?
Yeah, sure.
Whilst Catherine asks you something. So, rather than having as I said earlier, you're just a big walking tube.
I can just keep
And that sounds quite mean. But importantly, it's not a tube from there straight to your bum. If that was, you wouldn't have time to digest everything. You'd have your burger or your salad and you'd like, that's nice. That would be rubbish. It'd be a really rubbish evolutionary thing because it would be too quick. So instead, over the millions of years that us animals have been evolving, we've made everything slightly twisty and turny. Now, if you're driving somewhere or on a bike or on a run, it's only 1 kilometre between here and your home, you don't go, I'm going to make that a 25-kilometre run. Because that'd be really silly. It's the sort of thing I do, and I get really tired legs. But as food, you want to make that journey as long as possible. Why do you want to make that journey from mouth to bum as long as is physically possible without slowing you down? Why do you want it to be long? Yes, go on then.
So you have time to go to the toilet.
So you have time to go to the toilet? That is true. You go to pray, you're like, oh, that's nice. Oh, no, not again. Mop. No, I like that answer, but that's not quite right. Why might you need longer to have that digestive
Can I
tract.
just interrupt for
You
1 second?
can.
I'm about to break this so that it's one long tube, but I just want you to see how beautifully it sits when it's inside the body. So that every bit of the intestines has a nerve supply and has a blood supply and has a lymph supply, so that it can keep going, so it's got energy, so that if there's any bacteria, they can be treated with. So yeah, really beautiful structure. Here you go, look. It's almost butterfly-like. In fact, can I have 2 more hands to do the bottom bit?
Which bit?
This bottom bit here, so we can hold it out and they can see the full.
Where do you want this bit here?
Yeah. It's pretty, isn't it? Right, so I'm now going to turn that just into a tube, but that's how it sits when it's in the body.
Yes.
Carry on, Ben.
Yes, perfect answer. By being twisty and turny and all stuffed together, it allows time and enough space to take as many or as much of the nutrient content out as possible. Brilliant. You lot are great. So Jess, as she just said, is stripping away this connective tissue, this mesentery, which gives the blood supply, which gives the nerves to each little bit. And you can see it's almost like she's stripping out one little long sausage now. And this is the tube. This is what makes you you. This is your tube from one end to another. But as we saw, you can't just have everything. It's not just like stuffing a sleeping bag into a sack. You can't just shove it in there. It's really important. It's got its blood supply, it's got its nerves, it's got this whole organized system for things going in a certain place. And actually, Jess, I guess why can't you just shove everything into there? There's a risk of it being twisted and turned, I guess, right?
Absolutely, definitely. You don't want it to tangle itself up. I mean, you know, if you constrict one bit of it, the blood supply will be lost, and also the food won't be able to get through. I mean, that's disaster stations. That's the last thing you want. You don't want anything. You want everything to stay where it is, which is what all this stuff is about. Its main function, I mean, it's got blood supply and it's got lymph supply and it's got nerves, but its main function is like a hammock to keep everything in its little compartment to stay where it should be. I mean, there's more of that than there is of organ. So
it's really important, as we said, it's not just shoved in. So if you draw a body that you can see through, there's a tendency just to go squiggles. They're the guts. You couldn't be further from the truth. So Jess will know exactly which bit is where, even without being able to look inside, because they're in such a particular order. Yes, a couple more questions. Sharp top. Will you show the heart?
yeah,
yes.
we'll get to the thorax
All in
next.
good time. And yep.
What is the time?
Yes, there are different chambers. It's not all just, it doesn't just go at the same rate all the way through. So when you eat, your esophagus, as Jess was saying earlier, is this nice soft tube, pushes through muscular contraction, pushes down. The first big stop is, where does the food stop first of all? In the stomach. And that's where a lot of the breakdown happens, a lot of the digestion is going on, it's breaking it down to its smaller parts, and then we're seeing it in different speeds, different rates, different durations that it's hanging around doing its thing, and each time something different is happening. So maybe the nutrients have been taken out, maybe some of the water's being taken out, the moisture, and it's not just the same rate all the way through there. Yep.
Where's your esophagus?
Shark top. You had your hand up. It's all right. I know where you are, don't worry.
Right.
So already, when Jess is cleaning away this connective tissue, this mesentery, we're seeing that there's not actually that much in here.
That's all connective tissue.
That's all, yeah, that's all connective tissue, but really important. And then this is the tube. This is the thing that makes you, you. Go on, Em. We might do if we have time. As Jess said right at the start, because it's been frozen, we lose a lot of the structure of the eye, so it's just a bit of a ball of gloop now, unfortunately. If you're going to do the eyes, you do them really quickly, or you take them out and store them and then do them later. But at the moment, it's not very easy to do an eye that's been frozen and defrosted, sadly. Go on then. Yeah. It wouldn't melt your skin, but it wouldn't be great. So it wouldn't melt through, and it would just like an alien's blood sort of thing. But it would give you a burn. It's strong enough, the acid in your stomach, to give you an actual chemical burn. And you think about the stuff that's going on in there, and that it's not burning the inside of your stomach because of the special lining and all these things. But sometimes when you get older a little bit as well, you get some sort of acid burps and sort of acid feedback. That's when the balance isn't quite right in your tummy sometimes. But usually there shouldn't be a problem. But it is quite strong acid. 'Cause it needs to be. To turn something from a solid into a digestible liquid takes a lot of work there. Yeah. What's the green bit?
That. We're going to do this bit first, and then we'll get onto that. And I will tell you. Looks beautiful. Looks really good. So, this is the digestive tract. Ben has the anus.
There's a quote from Norwich Science Festival 2023.
I have the esophagus. So do you remember I put a cable tie through here around the esophagus to stop whatever was in the stomach coming out? So in the mouth, down the esophagus to here, about here, that's where I cut it off. So a little bit longer tube. This is the first organ it gets to, so what do you think this is? Yeah? Stomach, yeah. So this bit's the stomach. Something interesting in there. We'll have a look and see what that is in a minute. And then this is all intestine. And now that we've taken the connective tissue apart, it's really hard to tell the different bits. And actually, in an animal like this, where the digestive tract is a lot more simple compared to a herbivore like the llama we saw last year, less important, but different functions happening in different parts of the intestines. But basically, that's where all your goodness and your water is being reabsorbed. Now, does anyone remember how long the llama intestine was last year? I think it was longer than this room, wasn't it? It went a couple of times, whereas actually, this is pretty short, pretty unremarkable digestive tract in comparison. And the stomach, so the llama had 3 huge stomachs. And actually, this has quite a small stomach. So would you like to look inside? See what we've got inside there? So, oh yeah, measure it, perfect.
Measure, yeah. I don't want to hold the bum for too long.
Come this way. 3.5 meters plus probably 30 centimeters there. So 3 meters 80.
380, yeah.
Cool.
And this is fed back to people like Andrew Kitchener, the guy we were working with up in Scotland last year, who wants to look at the evolution of gut length in some of the big cats as well. Because it's a trade-off. You want a fast animal that doesn't walk around with a huge tummy, is not going to be able to chase its animal through the forest, yet it needs to be long enough to still do its digestive functions as well. But as Jess said, if it's not digesting things that are heavily fibrous, like lots of plant material, then it doesn't need to be as long. So it's always evolution's always a trade-off.
Now, one of the reasons we want to look at the inside of the stomach in a post-mortem is because we want to see if there's any ulceration, something that quite often happens in older animals. So, Ben, I'll leave you the insides, the gut material, the ingesta, and I'll just check the surface of the. Do we have any water? Yeah, perfect.
It's not poo, so it's the stuff in your. Oh, food. Sorry, yeah, it's food. So there's a big ball of hair here that we can see. Where are we? Can we see that? There we are. So you can see there's a big ball of hair in my hand. Now they didn't give this animal a big ball of hair to eat, obviously, but we guess it was given something that was hairy. So already we know what sort of animal will this be. A rodent maybe, or something like that. It's quite big for a rodent. Maybe a rabbit. So possibly something like a rodent, possibly something like a lagomorph, so one of the rabbits or hares. It's really hard to tell this sort of hair right now. And what's really interesting, there are no bones in here. So potentially we would look at this and say, well, either the animal's dissolved the bones, or it's licking and taking the meat off the animal there. I think, is that something? It is a bit like an owl pellet. As Jess said earlier, these don't really regurgitate things once they're in the stomach. Jess, I have found something.
Ooh, what have you found?
If you look there, there is something
Can you just get a bit more blue towel, sorry.
that is definitely not meant to be in there.
Oh, wow.
Sacking. And more.
Been eating something it shouldn't.
Now,
This?
what we've found in here is interesting, actually. We have a little piece of sacking in here that might have been used on the edge of its enclosure, that might have been used for part of its bedding, but there's some sacking in here that doesn't look like it's been there very long, actually. It's not heavily digested. It probably hasn't been sat there for days, weeks, months, or anything like that. And it
No, and there's no damage to the inside of the stomach, so I don't think it was causing a problem.
So we often see animals will swallow stones for different reasons, or they'll swallow different things for different reasons, but there's definitely some matting or sacking inside the stomach lining here. So you can see a lot of this, not a lot, but I guess about half of this is hair, and about a third of it is a little bit of matting here, or sacking, or something like that. Other than that, there's nothing else in there that's really obvious. There's no meat left, so it's been digesting its food. There's no bones in there. There are no little stones to aid digestion. So Jess will have to put that in the report. But interestingly, what's really nice is it hasn't caused any ulceration. It hasn't caused any lesions. It hasn't caused a blockage that could have caused damage further down the line. So that'll be what we call an incidental finding.
Yep.
It's all part of this process. But now, in the same way that we saw the scent glands earlier, yes, they were slightly different. Yes, they were slightly larger than we expected, or maybe expected. But actually, are they causing a problem? No. Is that normal? No, it shouldn't be there. Is it causing a problem? No. But it might allow zoos and other collections to go, really be careful, because maybe it was just this one ocelot. Maybe it just felt a bit hungry. Maybe it was just trying to have that one last meal. Maybe this is something that ocelots do. And if we saw this in every ocelot, we'd have to make sure that the way we look after them was slightly different. So this is why what we're doing today is so important for captive care. Right, what are we up to?
So this is the inside of the stomach. You can see it's not completely smooth. It's got ridges, and that increases its surface area. But actually, compared to a herbivore, or like the llama that we saw last year, it's actually quite a basic stomach. I can see that it's really, once you stretch it out, really beautifully smooth, and there's no damage there. So it had no stomach ulcers. Stomach was probably working as it should. And then so you can see that's the content that was inside the stomach. As we move down the intestine, you can see really quickly it turns from sort of something solid to a lot more liquid. And this is where a lot of the water reabsorption is going to happen. So it should start to become a bit drier again as it gets towards the colon. Although, to be honest, this digestive tract is pretty empty. What's that there? So quite often you'll see lumps of what essentially look like lumps of poo by the time you get to the end of the digestive tract. But this is quite soft all the way through. I wonder if it might have had a bit of diarrhea.
I've just been asked how long is a human intestine. I really should know this, and I don't.
I do not treat people.
Okay. I think it's about 7 meters. I might be wrong in that. Which is 7 big adult steps, and that's just the intestines. If you, as a little bit bigger than you, maybe 8 or maybe even 9 years, but 8 or 9 big steps like that is roughly how long your intestines are. Not much. Well, it'd be bigger than the ocelot, but it wouldn't be much bigger than the jag. You'd be surprised. The large intestine is, but it wouldn't be quite as narrow as this. Because we're primates and we've evolved to eat up in trees with heavily digestible, hard to digest material like leaves and fruits and things like this, we need a longer, more robust digestive system. So cats are famous for having live fast, die young, everything's streamlined the skull, streamline the teeth, streamline the guts and all this. Everything is as small as it can be to fit in. It's like a little sports car. You're looking all suave and cool and fast and sleek. You cut everything down you don't need there. So by cutting down this digestive tract, it's as small, as thin and narrow, short as it can be, and enabling it still to do its job. But like I said, Andrew Kitchener, the guy up in Edinburgh, is specifically looking at gut length in cats to understand how it evolved so quickly and so short. You compare it to the dogs, the hyenas, other groups, it's disproportionately much shorter than it should be. Yes?
Are you going to do it with the Jaguar's fur?
What are we going to do with the Jaguar's fur? We won't be saving the Jaguar's fur because it's already been defrosted a few times. It looks like it's in great condition. We wouldn't use this for anything now. So we don't use this for teaching. We might store a little sample just to look for coloration. Maybe a student will look at that at some point. But actually, if you store fur, hair of animals like this, you've got to have special permits. You've got to have a really good reason to do it. And because we don't work with these and no one else needs them that we know, we won't store them. So the whole thing will be disposed of in a very safe and way that no one else can take advantage of that, but we will be keeping the bones. So the skull, the teeth, the rest of the body, the skeleton is ours.
Okay. Oh, no.
How long has it been dead? So both of these been dead for over a year. So we've had both of them in our freezers now for well over a year. This has been out before, obviously, to have the post-mortem, which we did for our radio series. And for the post-mortem that Jess had to do. But the Ocelot has been waiting for today, basically. Good. Yeah, so the first series came out last year and the second series came out earlier, well, end of last year.
I know.
Hopefully, another one soon. That's the plan. So we're looking for a few more weird and wonderful things at the moment. Oh, what have we got?
So, does anybody know what this is? Liver, yeah. So this is the liver, really super important organ. Anyone got any idea what the liver does? Yeah? Cleans, yeah? I mean, you've got a lot of things that you can guess because it has like 500 functions or something. So, do something to the blood, yeah? It does, yeah. The blood goes to it from the digestive system. So, yeah, good. So digestion starts in the stomach where there's acids and things like that. That's sort of the breakdown of the bigger parts into smaller molecules. The liver also has a function in breaking down sort of proteins and fats. There's this little organ here. Anyone got any idea what that is?
Go on then.
The gallbladder. Yeah, brilliant. And does anyone know what the gallbladder produces? But it can have stones, yeah, you can get gallstones. And someone said it over there, bile, yeah, the gallbladder produces bile, which emulsifies fats and is really useful in digestion. The liver has a role, definitely, in breaking down proteins and fats. It's also the detoxifier. So if anything comes into the body that shouldn't be in the body, that needs breaking down, that's the liver's job. And yes, I'm just getting a bit conscious of time. How much time have we got left? Yeah, we've got quite a lot of organs to get through. So if you've got any questions about the liver, I can answer them, but I'm also concerned that we've got about 6 other things to look at. You can see it's beautifully lobed.
Does the tube go right through it?
The
The digestive tube.
The intestine?
No. So it's sort of, it was attached to it by connective tissue, but the way that the molecules from the digestion get into it is by a vein, the hepatic portal vein. So it brings blood from the digestive system directly to the liver. We can probably see the end of that vein somewhere. Oh, actually, I just need to cut into this and
In its most simplistic terms, when we teach at a much younger level, we say it's the sorting office. It's like your big regional sorting office where everything goes through there. You decide where it goes, whether it's good, bad, keep it, don't keep it, chuck it. That's the. I still think of that. It's, of all, as Jess said, it's so complex in what it does and what it doesn't do. If you just think it's sorting everything, that's where everything's decided. Good, bad, ugly, keep it, don't keep it. Which is why it causes problems when we abuse our livers. It goes through.
Where's the spleen gone?
Where's the what?
Spleen gone. Oh, there it is.
In the wild, we wouldn't expect to see it here. You often get a lot of parasites living in the liver. So things like flukes and other worm-like organisms sit in there. And we've seen between us a lot of parasites in livers. And Jess is doing a whole just regional cuts across at random, well, semi-random points.
It's gorgeous.
To look at the
Really nice.
to look at the internal structure and the colour. To look at the, you expect this uniform distribution of the material in there. If you're starting to see ulcers and little lesions, that's a bad sign. If you're starting to see capsules where you've got parasites living in there, also a bad sign. What we want to see, and Jess just said it's gorgeous, when you cut into that, it should all look really lovely, what we call uniform. It shouldn't be different colours and different textures. It should all look roughly, with your eye, roughly the same here. And that is a lovely liver, isn't it?
It's gorgeous, yeah.
Gorgeous, as Jess says.
It's fine. Quickly get that in the bin before it goes over.
Yep, hand up over there.
If you didn't know already, could you tell the age of the animal from inspecting the organ?
Oh, that's a good question. If we didn't know this animal was 15, by you doing what you're doing now, how diagnostic is it of age, looking at the internal organs like this?
I mean, his organs look great. So normally I'd expect, especially the kidneys as well, to look pretty bad in a 15-year-old cat, but it would be quite difficult, I think, if you were just given the organs. Yeah.
I mean, sometimes, yeah, sometimes no. If you look inside and they're really old and it looks frazzled, you go, yeah, it's a very old animal by looking internally.
And obviously lifestyle has a big part on it as well, and husbandry and...
Is that an offer for next year? We had an ostrich, we had an alligator. Next year, one of our regulars. Oh, yeah. This one here? What is the red bit you're looking at there?
The spleen. So that's part of the immune system. It's got two parts, red pulp and white pulp. Stores blood. To be honest, if it's big, we're worried, but that looks...
Why would you be worried if it's
lumps in
big?
it.
What does it mean if it's big?
Well, it's not working. I mean, that's the size it should be. So
Yeah.
if it's engorged, we've got to find out why it's not draining, or why it's inflamed, or why there's an infection going on.
So the spleen responds to infection really quickly, doesn't it? So
Yeah.
if you see a massive spleen, if we're sick and we're very ill, so I was quite ill before Christmas with an infection with a virus, I think my spleen would have been much larger than it should have been.
Right.
And had I been on Jess's table, I'd have been quite upset. But to be honest, my spleen would have been enlarged. So it's a way of looking at infection. So again, Jess says, even if it's normal, we still need to look at that. And what's normal for an ocelot might be normal for a different cat of this size. So again, it's understanding where we sit here. Yes? Oh, hang on, the lady behind you.
Do you find tumours in wild animals?
Do we find tumours in wild animals?
Yeah, wild animals, domestic animals, yeah.
Yeah, so tumours, so cancers and tumours are naturally occurring things. We did a dissection up in Cheltenham Science Festival a few years ago with John Hutchinson from the Royal Veterinary College and some other pathologists. They found a new type of cancer in cheetahs as we were doing the dissection that had never been seen before. And this whole team of pathologists absolutely lost their cool because they'd found this brand new thing that had never been seen. And the audience are relatively interested, but this was brand new for them. So we do see tumours in wild animals all the time.
So tumours are not to do with our lifestyle.
Some will be, but there are literally thousands of different types of tumours, of cancers, of different things. We're seeing loads more tumours in whales, for example, at the moment because they're exposed to sunlight more because they're having to come up to find more food. So there are lifestyle, and they're eating plastic. So yeah, there are things that affect tumour formation and types of tumours. However, they are natural through the animal kingdom. Some more so, but yeah, every mammal at least nearly every mammal at least will develop tumours. Certain species or certain groups like the naked mole rats are really cancer resistant. So there's a team in Queen Mary's in London who are doing a lot of work ethical work with naked mole rats to see why they don't get tumours, why they live for so long and why they're so healthy. So weirdly, cancer research often looks at other animals as well and post-mortems and research with live animals to see how we can understand stuff with us. So even though we've got a cat here, there might be human connotations or benefits or research down the line as well. Yes, question over there.
Whales be getting more cancers to do with microplastics?
Partly, it might be microplastics. Depends what sort of cancers we're seeing. So we are seeing some internal cancers and tumours in whales that are increasing. More importantly, they're spending more time at the surface because there's less food. And very often the acoustics are quite tough underwater, so they're being pushed to the surface more. Ozone's changing, so we're seeing more intense sunlight, so you're seeing whales coming more to the surface, and then you're seeing increased, and literally you are seeing skin cancer in things like blue whales more so. So there are teams working with skin cancers in whales right now. And we see in some populations up to 90% of the whales are developing tumours of one way, shape or form because they're spending more time at the surface. Again, another area of ongoing research. You never quite know where the research is going to end up, but that's another area. Yeah?
Do you dissect marine animals at UK or mostly terrestrial?
We will dissect anything that comes on our table. So we have sunfish in our freezer at the moment. We have sunfish. These big, they're great. But so, for example, the sunfish is really nice. We've had 4 or 5 on the Norfolk coast the last year, 2 years now. We don't know why. We got a team in from down the southwest who are world experts in that to see if they need anything, if we can help them. So we do whatever washes up, we'll take. We're not fussy. And the nice thing is we'll collaborate. So we had earlier Johnny and the rest of his team who are taking samples from this cat. We had another team doing the sunfish stuff with us. So it's, if we can't do the stuff ourselves, we'll bring different people in and try and link in students and academics and others to try and... Are you at UEA already? Or?
Yeah.
One of mine? Haven't seen you yet, so okay. Okay, cool. We have stuff in bio.
I might do evolution biology next year as well.
See you next year. Yes. Oh, hang on. He's got a question. Is someone? No. Yep. Oh, yeah.
Yeah.
Is there much difference in carnivores and the size of the
Is there much difference between herbivore and carnivore in terms of gallbladder size and form and function?
Hang on 1 second. Yes, is the question. And some animals don't have gallbladders, and some bile empties continuously. But in terms of function, it's always for emulsifying fats. And obviously, sorry, I'm not very good at multitasking. Obviously, some animals have a much fattier diet than others. So for a herbivore that's eating mainly plants, mainly grass, I mean, there's not much fat.
Yes, there are differences. I mean, Jess is doing incredibly delicate work. It looks like she's just hoofing a scalpel in there, but to actually get, I mean, Jess is absolutely amazing. We spent well over an hour once looking for one ligament to find a jaguar, in that jaguar's throat, enabled us to find where the roar came from. So what she's doing is incredibly delicate. And I'm being very unfair asking her to multitask here. Yep. Why is she not cut out some of the organs?
Which organs?
Why are the kidneys still in? Why are they not all out at once?
I was about to ask them what they are.
Oh, right. Well, he did it. He ruined it for me. I got blamed
They're
because
still
of
there
you.
because I was going to ask them.
It's
What do
his
you
fault.
think these paired organs here are? Thanks, Ben.
So you got me in trouble now.
So we've got some kidneys here.
Next year, you're doing this, all right?
Yes. So these paired organs here are, as Ben kindly described for me, kidneys. They have got these tubes here called ureters, and so you can see that tube there runs all the way down here. So what do you think this is? So the kidneys are filtering the blood and producing urine. So what do you think this thing here is that their tube's going to? Bladder, yes. So it looks like a small little sac, but it's super, super. What property do you think it has to have? Has to be super stretchy, yeah. So, I mean, that doesn't look like much, does it? But if you think about how much of a wee your dog does when it goes, I mean, sometimes they do these little wees, but if they do a big wee, yeah. Let's open that a bit more.
thread and a needle?
Where are the... For them. Hang on, that is going to... Not really, it's quite slippery. The thing is, obviously, it's a closed system normally, so... But you can see that's got loads of stretch in it. I'm tempting fate there, so I'm not going to keep doing it, but trust me that it's very, very stretchy. Now I just need to check the inside of that, and then we'll check the inside of the kidneys. Has anyone got any idea what this here might be? So it's sitting, it was sitting right beneath the bladder. Only males have it. Yeah. Its penis is here. Its testicles were here. Ben, any idea?
What's the question?
What this might be. So the bladder was sat here. This was the penis. The testicles were here. Only males have it.
Only boys have it. Begin with P?
Yeah.
Do you want me to tell them?
Yeah.
I'm terrified of
It's the
answering now.
prostate.
You are going about the wrong way of not volunteering for next year.
Just looking at the inside surface of the bladder. And then, how much time have we got left?
20 minutes.
20 minutes, right,
So
right,
each
right, right.
time Jess is looking at the external colour and shape and size of these organs, she's looking internal, if it's possible to look at these, either slicing through in sections to look as cross-section, or she's looking at the internal anatomy of some of these little sac-like organs.
So, it's really hard to show you, but you can kind of see here. Can you see that faint white line there? Not that one. Oh, it's really difficult to see. Basically, there are tiny, tiny bones in here. I would dissect them out, but it's going to take me ages and we won't get to see everything else. But basically, they're really small, delicate bones. And they are what allow small cats to purr. So when they vibrate their larynx, these tiny little bones that go between the base of the tongue and the larynx here vibrate, and that's what makes the purring sound. In the Jaguar and some of the other big cats, that has been modified into a really tough, strong ligament which can pull the larynx right down. And that allows them to make a noise that no other animal can make, which is, what noise do lions make? A roar. So that's why these big cats can roar. They can pull their larynx right down with this strong ligament, make this huge, loud sound. So an ocelot can't roar, but the trade-off for that is it can purr because it's still got those thin hyoid bones that can vibrate like that. And because of that, big cats like lions and jaguars and tigers can't purr, but they can roar.
So if you've got your cats at home, or Granny or Grandad got their cats, when they're purring next time, just gently put your finger at the base of their throat and you'll feel that that purring. As Jess said, you can feel it vibrating. Just if you gently put your fingers on there. And again, it's a way to communicate. It's the same bones, the same structure, but slight modifications, as Jess said, allow them to do very different things. You wouldn't want an ocelot roaring in the middle of the jungle. It's going to get in trouble straight away. But if you're much bigger and you're stockier and chonkier, like a jaguar, it's okay to roar. Not many things are going to pick on a jaguar. So Jess is moving down with this weird tube-like structure, which looks a little bit like the cable of a Hoover, like Henry the Hoover there. Oh, wait, what? So Jess is now taking what we call the pluck out, or the top part of the pluck, which is the tongue and the rest of that throat section. So we've got to do it in a set way, that it comes out looking nice, but also in one structure that we can look at. This is next to this, is next to this.
But
which
normally I
allows
could
Jess
open
to,
this and
yeah.
pull it, so it might not be as beautiful as, I feel like you're setting me up to fail there.
So I'm making Jess's job much harder today, because usually she'd have a set of bone cutters and we'd go straight through these ribs. However, that ruins my bones. So I've asked her to do a very difficult job and take the whole thing out by not cutting any bones, which shows just how mean I am, but also how good Jess is at her job. But yeah, taking everything out in the right order is a way to allow Jess to standardise what she's looking at. Yep?
Basically,
Can you see the heart?
That's what I'm going to be getting.
That's going to come out with the rest of the pluck, the rest of this thoracic section, from tongue down to heart, lungs. The whole thing will, in the same way that Jess took the digestive tract out in one nice bit. You got the same thing with the thoracic, the respiratory section, circulatory section.
I don't want to cut, but I want to try and separate that. Yeah, I'll need to
we're going to show you the kidneys, we're going to show you the heart, the lungs, but like I said, Jess is going to take this section out first, and then she'll go from up here and take that section out, and the whole thing will come out as one weird-looking structure.
Ben, do you want to try and, well, I just don't want to break the hyoid. Are you bothered about them?
No, go for it.
Okay.
It's not a very good question.
It's all right.
You know when some animals are really smelly, have any of them ever actually thrown up?
Have we ever had I like this question. Have we ever had anyone throw up at one of our dissections? We haven't, have we?
I don't think so.
Don't think so. I think if you're in here long enough, you're self-selecting to be slightly weird, you lot. And it's good. We're all weird. Everyone behind here is quite weird. Both weird? Yeah. These guys, definitely weird. Luckily, we've never had anyone be sick in here. And partly that's a nice reflection on you guys that you're all in here and you're all happy to do this. But equally, I think once you get in here and you see this, the idea of, we tell people, we're doing a dissection, we've got this and this and this, it sounds gross. But actually, I mean, some of us are veggies, some of us are vegans up here, some of us are meat eaters. But it doesn't really matter, because what we're doing is, we're seeing this in a way that it's the science, and that's, I think, really helps. And we're talking about the science, we're not making it gross for you, we're not talking about this and that and the other, we're normalizing it. And what we're doing, luckily, we've never had anyone be sick. I've been around people who've fainted before, in anatomy work. Never a kid, always the big roughy tufty blokes, I hate to say it, who go down like a sack of spuds.
Whoops. Ben, are you happy to talk about these two while I try and go in and get them?
So Jess has managed to put a couple of structures here. So sorry, I just pull it like this. So when you eat your food, which tube do you want it to go down? It sounds a really silly question. Yes. The esophagus. Yes. Which tube do you not want it to go down? The trachea, the windpipe. Yep. And that's a really big risk. Now, sometimes people say to me, is evolution random? Has someone designed evolution? And one of my answers is very often no. Because whoever designed evolution, if there was some greater being in my mind, wouldn't put the two holes next to each other, where if the food goes a millimetre in the wrong direction, it goes down into your trachea and you choke to death. Yeah, it's not a good design in any way, shape or form, and yet there's a little flap that, just like a bin lid, that just stops the food going down the wrong hole. If that goes wrong, we're in big trouble. And we saw this, didn't we, with the porpoise last year? A big cause of death of porpoises and dolphins in the UK, especially porpoises, are flatfish getting caught at the top of the trachea and unfolding and sitting there. And because the tubes are next to, the openings, the tubes are next to one another, Rob DeVille and his team at ZSL, Zoological Society of London, not often, but semi-often, will come across something that looked healthy, looked happy, get inside, get to the trachea, and you're like, oh, there's a flatfish. And it just swallows, doing its thing. They haven't got knives and forks, haven't got teeth to chop things up. They swallow these fish, they unravel, and they sit at the trachea, and they choke to death underwater. It's a horrible way to go, but it shows how dangerous these things could be. Now, a lovely adaptation are the two different types of tubes here. You've got this really soft, wiggly tube that you can barely see. This is the esophagus, and it's like this sort of soft tube that sits from mouth down to the stomach. This other one, this amazing tube with these little loops around. What do you reckon these loops are made of, anyone? You can feel them in your throat. This tube that you can feel it. Don't squeeze too hard. You'll cause yourself some damage. But this tube here is exactly the same as what we're seeing in here. What are these little rings made of? Cartilage. And where have you got cartilage in your bodies? Anyone? Yep.
Inside our bones.
Not inside your bones, between your bones you've got cartilage, yeah? Where else? Yep. Joints, so your joints have nice cartilage linings, and if you get bad knees and you get to our age sometimes, it's a cartilage wearing down. Where else might you have cartilage in your bodies? A really obvious place, yes, your noses, nice and flexible, wiggly noses, and your ears. But you've also got them in your trachea. And keeping your trachea open is absolutely essential. So when you're swallowing your food and it goes down this tube, if you're not swallowing food, it doesn't really matter. What you can't do is have your windpipe close up. What will happen, sounds really obvious, you will stop breathing and everything stops. So to keep your windpipe open, it's really important to have this, what we call these cartilaginous rings here. So it's the same sort of material that we see in between our joints, in our noses, our ears. Amazing little adaptation there. How's it going?
It's going.
It's going, great. A nice thing for me is that we can see these huge balls on the side of its cheeks here. These are massive muscles. Now when you clench your jaws together and you slightly clench your teeth, you can feel them at the side of your mouth, yeah? Have a go. These are your masseter muscles. These are big muscles that are there for chewing. And having these big cheeks, not because they look cute, it's because they've got these big cheek muscles that are able to really push and power that slicing adaptation. So when we see these big skulls on our lion here, Siberian tiger, thank you, I was wrong with that one. When we see these big shearing teeth, it takes a huge amount of muscle. So these big arches that we see here, on the side, here, these zygomatic arches and the sagittal crest here, these are all for massive muscle attachments. So even though this tiger is a much bigger skull, we're still seeing the same sort of cheek morphology, these same massive muscles. They haven't got the big chomping molar teeth that we have. However, they've got these big shearing teeth that I keep mentioning that I love. And by having these big cheek muscles allows us to understand that they are powering through this meat. They're shearing stuff off. They're using the side of their teeth to take chunks of meat off their prey there.
Nearly there. Nearly there. What is that?
So you're looking up into the chest cavity here, so you're looking up that bit. So Jess has moved the diaphragm away. We're looking past the kidneys, and you've got that chest cavity where everything has nicely come out. Well done, Jess. And you've got what we call the pluck, which goes from the tongue into the trachea down to the heart and lungs.
I actually didn't get the tongue because I couldn't get it through the hole.
Okay, oh, the tongue's here, yeah.
The heart is in here, yeah. Now these lungs don't look great, but I've sort of grabbed and pulled at them a bit as I've been trying to get it out, so.
So remember, this thing's been in the freezer, so it's been frozen, it's been defrosted, so we will lose some of the quality of the organ there. But again, Jess is looking for lesions on there, she's looking for signs of decay, signs of infection. Also looking for the heart. You often see fat around the heart if there was a problem. You might see scarring around the heart in a larger animal. So again, Jess is constantly looking for different problems, different pathologies, different injuries around each of these organs each time. Anything obvious so far, Jess?
I mean, these lungs don't look great, but...
Can I move you just to the side slightly? That's all right.
I mean, I think that's partly because I've pulled them about, partly because it's been frozen, and also partly because it was given some medicine before, well, to kill it. I'm going to cut into them in a sec. So, this is the pluck here. Normally you'd have the tongue on there as well. This is the esophagus. This is the trachea, as Ben was just explaining to you. Now, cats have really, really complete rings. So in a dog, there's quite a wide bit at the back. Where the rings aren't complete. But these guys have quite a solid tube. Not really significant apart from when you're trying to put a tube down their throat. You have to be a lot more careful in a cat. So esophagus there. And you can see, obviously you want special flaps. I think Ben already talked about this, to prevent any food from going down there. So this is really, really sensitive. If it senses that anything's getting to the back of your throat, it wants to protect the top of that tube, so that flaps over. Right. I am aware that we're running out of time and I want to get through everything, so...
We have 10 minutes.
Okay. Is there anything else?
We've got about
After
10
this.
minutes before we're due to end, but we are coming towards the end of the post-mortem now. Quick recap. We've had a look through. We know this animal was 15 years old. We know it was from a zoo, and unfortunately, it was deemed to be arthritic, and its quality of life was declining. Jess, as a veterinary surgeon, is required by law, well, the zoo is required by law, to have a vet look through the body and to see if there's an obvious cause of death, if there's any sort of mismanagement, if there's any welfare issues. What have we found so far, Jess? Is there anything indicating an obvious cause of death there?
Had a bit of diarrhea. Was something else. What was the other thing? Oh, it had that in its stomach. Nothing major really. Right, so I've just cut through the membrane surrounding the heart. And
Yeah, what's your question? Can you cook a deer's heart and eat it? But can I or can you? Can anyone? Can anyone cook a deer's heart and eat it? Yeah, if you want. That could be one for next year. Cookery skills with Ben and Jess.
Vegetarian.
Yeah, you can. I mean, a lot of people eat venison. A lot of people, I mean, my granddad used to eat hearts of different animals. I didn't as a vegetarian. But yeah, a lot of people will. What you don't usually eat is predators. So very few things. If you look in the natural world, very few things eat predators. And when you get that, it's really special to see one predator eat another predator. We call that hyperpredation, but actually eating predators can lead to lots of infections, lots of parasites, lots of problems. So most things, you don't usually see a lion hunting a cheetah. If they do, they kill them for competition. They don't usually kill them for meat. It's really unusual to see that. It is, but there are occasions you see other predators will hunt predators. And like I said, that's called hyperpredation. Really unusual to see that. You see it in the marine ecosystem more, but very rarely with mammals. You get that build-up of
Let's say a meat is like, let's say a frog, it eats stuff, right? It eats insects. Or if there was an insect that was a meat eater, such as the frog.
Oh, you get it with other groups, so reptiles and amphibians, but you don't really see it in mammals. There aren't many examples that I can think of where one mammal will intentionally go and hunt another mammal and eat it, and it's not competition, it's not territorial behaviour. There's examples of like golden eagles eat foxes. That's called hyperpredation or mesopredation, but it's not an example of one mammal hunting another. Yeah, that's fine, but like I say, it's only with I can't think of many mammals that eat other mammals that are both predators. We see things like, well, okay, one of the few examples will be killer whales and seals or sea lions. So they'll do them, but there's not a massive build-up, but you don't usually see, there aren't many examples. Yeah, yeah. Yes? What was wrong with the jag? Do you remember?
Yeah.
Oh, yeah.
It'd gone off his legs, hadn't it?
So this was a very, very old Jaguar, and it was, you can see here, you can see where the bones are, you can see where the hips are. There's a real quick way to look at that and go, well, that's the zoo was bad, and again, I don't like zoos very much, but even me, I can't say that's because of a bad zoo. It's a very old animal, and like sometimes when we get old, our muscles disappear and we get harder to walk around. It was starting to lose muscle content on its back legs, and the decision, even though it was a healthy, happy, big boy, they could have pumped him full of medication and drugs and things, but actually sometimes for animals, the kind of thing to do was to put him to sleep. So the decision was made to put him down. When we did the post-mortem, there was nothing obvious that showed us the reason for that.
This is the heart. This is the heart. So this is the heart. Sorry. I'll talk about that in a second.
That's it. So a lot of people think that great whites are apex predators, but killer whales take them down. There's very few things that take a killer whale down. So an apex predator means there's nothing above it. And poor great whites have found out there is something above them. Killer whales. There's not many things that will take a killer whale on, though.
So what I have now is you're all just saying it, the heart, which is, like all mammals, a four-chambered organ. Anybody know the names of the chambers?
Someone said it. Yep, the ventricles.
Ventricles, right and left, and
Someone else just said it. Atrium.
Atria, right and left as well. So the ventricles are the ones that do all of the pumping, so they need to be big and strong. The left ventricle is what's sending blood to the whole of your body, apart from the lungs. So that needs to be the biggest, strongest part of the heart. That's this bit here that I've just cut through. And you can see that the walls of it are really, really super, super thick. Now the other ventricle needs to be quite strong because it's pumping as well, the right ventricle, but it's only pumping to one part of the body. Anybody know which part of the body that is? The lungs, exactly, because it needs to go there to get oxygen. So, I think I just need to change my gloves. So the walls of that are much, much less muscular. Because you don't want to be pumping blood at really, really high pressure through the lungs, because the lungs are really delicate organs. And if you're pushing anything at too high a pressure through there, okay. I'm going to do this quickly because we need to, what else do we need to do? Where have we got to? No, it's just kidneys, isn't it? Are we done?
Yep. So
Perfect.
we are winding down. We've only got, unfortunately, 5 minutes left before we have to close up. But as we've said throughout today, for those who have just joined us, not been from the start, this animal wasn't killed specifically for us today. It was an animal that lived in a zoo that was a very happy, healthy life. However, when it turned 15, the decision was made to euthanize it because its quality of life seemed to drop. It seemed to be arthritic, and there seemed to be a few health changes that we weren't, or that the zoo wasn't entirely certain of. And with Jess being a veterinary surgeon, she's required by law, or the zoo's required by law, to do a post-mortem to see why the animal died. Now, we've spent the last 3 hours now looking to see why this animal died, whether there's any infection or disease or injury, and so far we haven't found anything obvious that led to that. However, what Jess also made very clear is that by doing a dissection like this allows us to understand what these animals should be like. And we are one of the few, you are one of the few people in Britain to have seen inside an ocelot like this today. So this lovely little cat from South America is helping us understand just how these animals live in captivity and also potentially how they can be saved in the wild. Jess.
So we've got the two ventricles here. This is the left ventricle, which is much, much thicker, and that's the one that's pumping to the whole body. And this here you can see, I mean, it hangs differently because it's floppier. It's also muscular, but that's pumping just to the heart, just to the lungs, sorry. So you can see, I mean, just the one wall, there's a tiny, tiny little space in there, which would be stretched when there was blood in there, but each wall is about that thick, whereas this side, much flabbier, you can see. And then the atria are much smaller. I'm not going to dissect those because we don't have time, but there's one of them there, for example. And they're just for storing the blood. So the blood comes into the atria, goes through a valve into the ventricle where it is pushed out of the heart through a great vessel. We can come back to that if we have any more time, but yes.
Is a human heart bigger than a ocelot?
Yes, so a human heart is about the size of your fist. So you see that's quite a lot smaller, isn't it? But still quite a big organ compared to the size of the body. These are the lungs here. They're quite congested, but I would say that's normal for the way that it died. Normally you'd see sort of a nice butterfly shape, but it's been frozen. I've had to pull it out without opening the chest, so they don't look as beautiful as they should, but you can see that the trachea goes down here and then bifurcates, which means it goes into two. You see that there? One to each lung. And then, like the branches on a tree, the branches just get smaller and smaller. Can you see that starting to happen there? Sorry, I'm in the wrong place. Let me just quickly look inside the kidneys. Ben, can you ask their questions?
Not paying attention.
Could the reason it's like the song was stuck in, or was kind of a bit wedged but still was breathing a little bit here?
Well, we know that the reason that it died was that it was put to sleep because it was poorly. I think just generally its quality of life wasn't great, and that's why the decision was made probably, because there's nothing really specific that I've seen. It feels just like, if you feel a hoover, the tube that goes to sort of the sucky bit at the end. It honestly feels exactly like that.
Also, it's like the things that you can pull out and it makes a little popping sound.
Yeah, a bit.
The
Yeah.
plastic.
Question from up high.
Question, yes.
Is it time?
Yeah? No? All right then.
Last thing, inside of a kidney. So we're not going to have time to go into what each bit does, but first of all, when you cut it open, it looks like a beautiful butterfly. And also, it's got this cortex on the outside, the medulla, and then this is where all the vessels gather, and the tube would come out here, going down to the bladder. So this is where the urine is made. The urine goes through that hole there and down into the bladder.
How, you know water that you drink?
Yeah.
I've got to be very mean and say that is it from us today. We've had a lovely opportunity to investigate one of what I think, even though it's a small cat, is one of the most impressive predators of South America. And if you were anything smaller, or even the same size as this ocelot, you would not be safe walking around at night. These look like cute, cuddly little cats. They are ultimate killers. If Steve Backshaw was here, he'd be going, "This animal is absolutely amazing!" But he's not here, so you get me and Jess. I'm going to try and get Steve here next year, but let him know. As Jess has been saying all throughout today, this is an amazing opportunity to really investigate the science of what's going on. And there are different ways that we can look at the genetics that's going on at UEA to look at how animals reproduce, the welfare in captive situations to see whether we can keep them any better. And ultimately, the more we understand about these animals, the more Jess and her colleague can look after them in captivity, and the more we can hopefully help them in the wild. As ever, some of you guys have been here for for nearly 3.5, 4 hours, and I am, as ever, it's hard enough for us to do this, jumping around doing our thing, but the fact that you've been standing here, especially mums, dads, grandparents, and others, thank you for that. I really appreciate this. We couldn't do this without you, so thank you so much for supporting Norwich Science Festival. If you are happy with this, please let them know that you enjoyed this. What would you like next year? Yes. If you say unicorn, get out.
What about beaver?
A beaver? All right. A great white shark?
Might need a bigger tent.
We'll just try a unicorn then, it'll be easier. Yes.
Cheetah?
A cheetah. Okay, that's more doable. Yeah. A zebra.
That would be fun.
A cobra.
Probably get a cobra.
We get a cobra. Okay. Yep. A lion. Okay. A lion? 2 lions or the same lion? Same lion, all right, thank you. Yep. Alligator. Okay. Yep. An elephant.
I don't think we do that in 3
No,
hours.
you know what? Get her an elephant, and you're going to come help us next year with that, because you're going to be...
We'd end up inside it.
You'll have to get inside and be like, what's it like in there? You're like, dark. All right? We're going to try and get, as ever, we're going to try and get something weird and wonderful and exciting and interesting for you next year. A lot of this depends what we can get, but it's nice to know that you come back and come back every time. We nearly had a dolphin this year. We were trying to get a dolphin, although we've got to get a few more ticks in terms of our health and safety for dolphins. So thank you so much. We hope you've enjoyed being with us in this wonderful investigation. Can you just give Jess, Catherine, Emily and Ellen a round of applause? Thank you.
And Ben.
And our wonderful team here, our wonderful live streaming service. Right, we hope you enjoy the rest of the Science Festival. There are lots of other events that Jess has got going on, that I've got going on, and we hope you enjoy the rest of this week. See you throughout the week, and if not, see you next year.